and Agriculture Research is organized by Department of Biosciences collaboration with Research Foundation of India. As per our going to our core culture, as per Indian tradition, to begin with any good and noble work, we seek the blessing of Goddess Ma Saraswati. So before officially beginning of our session, we start our program with Saraswati Vanna. For that, I would like to invite Dr. Indira Dikshit ma'am from Department of Commerce. Indira Dikshit ma'am. I think she is not there. Indira ma'am. Now, I welcome Honorable Principal Dr. Rajesh Shah sir. I welcome head and faculty of various departments. I also welcome today's session all the students and participants from different places. I welcome Dr. Azmat Alam Khan from Jammu Kashmir. Welcome sir. Now I invite Shiv Om Pratap sir for their talk.
Ma'am, I think Indra ma'am has joined. So please.
Okay. Okay sir. Indra ma'am. Now I invite Dr. Dikshit sir to say a few words.
Indra ma'am, please
Yes, sir.
go for Bandana.
I am audible, sir.
Yeah.
Okay. Good morning, all of you. Jai Ma Saraswati. जय जय हे भगवती सुर भारती तव चरणों प्रणमाम जय जय हे भगवती सुर भारती तव चरणों प्रणमाम नाद ब्रह्ममयी जय वागीश्वरी नाद ब्रह्ममयी जय वागीश्वरी शरणं ते गक्षाम जय जय हे भगवती सुरभारती तव चरणों प्रणमाम त्वामसि शरणा त्रिभुवनधन्या त्वामसि शरण्या त्रिभुवनधन्या शरणं ते गक्षाम जय जय हे भगवती सुरभारती तव चरणों प्रणमाम नाद ब्रह्ममयी जय वागीश्वरी नाद ब्रह्ममयी जय वागीश्वरी शरणं ते गक्षाम जय जय हे भगवती सुरभारती तव चरणों प्रणमाम जय मां सरस्वती.
And thank you, ma'am. Thank you so much. Now I invite Dikshit sir to say a few words.
Good morning to all of you present over here in virtual meet in our international conference. Actually, right now, a slight change has been taken in today's session. Actually, this session was meant for Dr. Ajay Thakur sir from ICAR CPRI Simla. But due to unavoidable conditions, being in a meeting, Dr. Thakur sir will join us nearby 12. That's why I want to take this opportunity to present myself on my talk, and after my talk, Dr. Ajay Thakur sir will join the session as a eminent guest for today's invited talk. Right now, I am going to take this opportunity for deliver my talk. So I am just going to thank you, Ekta Madam, for giving me this opportunity to say few words on my talk. I am just going to share my screen. Am I audible to all of you?
Yes, sir.
Am, am I audible?
Yes, sir.
Yes, sir,
You
you
are
are
audible.
audible.
Please be continue.
My presentation is visible to all of you?
Yes, sir. It is visible.
Okay. Again, a very warm good morning to all of you. I am going to present my deliverance on the topic contemporary selection paradigms for enhancing qualitative and quantitative productive traits in crops. Basically, from the beginning of humanity, if we talk, we always found depend upon the plants for getting our daily meals, our furniture, our clothes, etc. from the beginning of the humanity. For this purpose, we have used breeding concept for enhancing our crops productivity by using various conventional techniques, just like when we are going to marry, our parents are going to select a best male and best female for going to marry, for producing very good offsprings in our future. जब हम शादी करना चाहते हैं तो हमारे जो पेरेंट्स हैं वो एक बहुत ही अच्छा मेल या फिर एक अच्छे बॉयज या फिर गर्ल्स को सेलेक्ट करते हैं। किसके बेसिस पर? उसका जॉब कैसा है, उसके अर्निंग कितना है, और गर्ल्स भी अगर जॉब में है तो ठीक है कि नहीं। और उसके बाद फिर एक न्यू फैमिली इज गोइंग टू स्टार्ट, और उसके बाद जो उनका प्रोजेनी रहेगा दैट विल बी मोर एफिशिएंट, that is called a generation to generation. In a same way, when we want to produce a crop or desired traits in our crop, then we have to select a male plant and female plant. After that, we are going to mate them for producing desired offspring. For example, we have inculcated disease resistance, we have inculcated genetic transformations, we have made certain modifications in our crops for getting maximum productivity. But with the increasing population, whatever efforts we have done always remain with certain drawbacks. That's why conventional breeding program have not produced productivity as we have expected. That's why I am going to talk on the modern selection paradigms. Simply, we can say the recent tools and technology for selecting a plant which have better genotypes, phenotypes, which can produce very good productive traits or productivity in our forthcoming generations. That's why I am here. My talk is contemporary selection paradigms, यानी कि वो recent tools which can enhance, जो कि हमारी पैदावार को बढ़ा सकते हैं on numbers में और nutritional values में, in our crops. And this paper is undergoing for publication also, under publication. Content. These are the contents on which I am going to talk with you people. Conventional agricultural approaches in India, which have used earlier till some time ago. Plant breeding regimens, which type of plant breeding we are going to utilize. What is the need of the study? Why I am here to talk on this talk? What is the relevance of that? Molecular breeding via mass. Either we can utilize recent tools like SNP, RAPD, microsatellite markers, etc. for performing molecular breeding. Molecular means when we are going to utilize certain recent biotechnological application, either genetic markers in terms of DNA markers or RAPD, RFLP, AFLP, etc. Or we are going to use biochemical markers in terms of enzymatic catalysis, etc. or phenotypic markers also. Advanced tools of biotechnology, what are the recent tools and techniques which can enhance the selection regimen for maximum production of our desired crops as well? What are the advantages of biotechnology for agricultural crops? And in the last, conclusion. If I want to talk on the history. Human depend upon the plants for their survival from the beginning of agricultural practices like furniture, nutritious food, fruits, etc. Indian farming system was carried out through conventional breeding program. Mainly, I have given you an example of marriage. Just we have to select our plants upon the basis of phenotypic traits like height, color of the flower, what is the एक just a simple phenomena how that is productive. Upon the basis of these states we are going to select the plants. But just friends, just you have to think, if we are going to select the plant or desired plant or host plant upon the basis of phenotypic traits which are going to affected by the environmental factors, then how can be able to produce our efficient cropping system? अगर हमारा सिलेक्शन ही पुअर रहेगा, फिनोटिपिक बेस पर रहेगा, जो कि इन्वायरमेंटल फैक्टर से अफेक्टेड होते हैं. जस्ट सपोज टुडे heavy raining is there, heavy raining is there, heavy temperature increasing is there, or which may be affect our crops, but we have selected our desired plant upon the basis of these environmental factors which are not found sure, then how we can produce an effective crop? We have also used organic farming, was utilized from biological based using conventional seeds, systematic progression of cropping cycles as well. We are very well known if we have 3 or 4 cycles of the crops in a season or in a year, 3 or 4 seasons for wheat, for barley, for rice, etc. upon the basis of temperature compatibility. Which type of temperature our crop want, that is going to crop cycles. Now, even adverse environmental conditions, as I have told you earlier, like extreme hot, dry, and raining, which may cause more loss to our cropping yield and make conditions even more worsening in terms of productivity, in terms of growth, in terms of metabolic pathways. To meet with the required nutritional food, various revolutions have been employed yet. For example, Green Revolution, and Dr. S.C. Maheshwari was the father of revolution, Green Revolution, for elevated yield of cereals like soybean, maize, wheat, etc. through Green Revolution. Yellow Revolution has employed to attain the maximum productivity of oil seeds like rapeseed mustard, yellow mustard, etc. Operation Flood to enhance the productivity of our milk by modification our livestock in terms of Murra Gai and certain import species like Jersey, etc. Willy Revolution for fishery purposes. Fishery means we have just introduced the genetic engineering, we have just introduced molecular breeding for hatching our fish with higher body weight, more productive traits and growth for fulfillment the need of food in terms of meat. That's why value revolution was attained by the central government or ICR to enhance our either cereals, oil seeds, dairy products, which is still going on, and fishery products as per our need. Now, plant breeding is a art and science. Anybody can interact with me. I have used two words, art and science. It may be art or maybe science of changing the traits of plants in order to produce desired characteristics was started back 9,000 years ago. Breeding is a art because we are just going to plan to mate two individuals, while science is it in a way because it is going to follow the Mendelian laws in terms of genetics. Clear? That's why breeding is always remain as an art and science of changing the desired traits which we want, like disease resistance, productivity, growth, metabolism, etc. The science of plant breeding was further developed in the 20th century, and plant breeders very well understood how they can select the superior plants upon the basis of phenotypic traits only, not upon the basis of genesis. The main objective of breeding was remained to improve the quality and quantity. Quality means we are just going to enhance the just we are phenotypic traits, and quantity means we are just going to enhance the number of traits like productivity, nutritional, abiotic stresses, and disease resistance in our plants, etc. Just these four was the main objective behind the breeding program, which was started in nearby 1980s and 1990s. Just believe me, that was the molecular revolution. Just believe me. Now, since the practices of agriculture began in the 8 to 10,000 years ago, farmers even could not have about the genetic makeup. Just you can believe if we were utilizing certain crop cycles from last 10,000 years, but still in the villages, the farmers don't know about the genes, don't know about the genetic makeup, don't know about the disease resistance. They just select the best plant upon the basis of phenotypic traits only. This plant have a this height, it may be better. This plant has produced such type of cereals physiologically, morphologically. Then they have select. This was the conventional system in which we have selected our desired plant upon the basis of our just phenotypic trait like color, seed, morphology, height, etc. Farmers usually selected the best plant and seeds upon the basis of morphological basis, as I have told you. Then, once the science of genetics have inculcated through Mendelian genetics, then we become aware about the genetic makeup of a plant. We know, we try to know that the phenotypes is always depend upon the genotypes. That was the revolution by which our crops become more efficient through in 1990 and up to 2020. Upon the basis of that, we have inculcated the principle of genetics, that we know if our parental species are found efficient, then their progeny will be efficient. Just in a way, the children's of a IAS, PCS, or doctor always will be a scientist, doctor, or PCS. In the same way, that's why our farmers or we have used the certain thoughts, if parental parent remain more efficient in terms of plants, then their progeny will also be more beneficial. Now, after the knowledge of genetics, we have started classical plant breeding as compared to conventional plant breeding, which the conventional plant breeding was depend only on the phenotypes or phenotypic trait selections, while the classical plant breeding programs remained upon the genetics, the knowledge of genetics, which was inculcated through Mendelian age. Various in vitro techniques such as protoplast fusion, in which we have to select the host plant, we have to collect protoplast from their leaf. Protoplast means the cell without cell wall. We have just degraded the cell wall, we have just taken the plasma membrane for fusion, for crossing, the embryo rescue, etc. These are the certain techniques which were inculcated during classical plant breeding for making our crop more productive upon the basis of genetic diversity, hybrid plants. I think all of you have learned or listened about the hybrid seeds. Hybrid plants are nothing. These are the combination of two best parental traits of parental plants. If I want to give an example, today we are going to utilize mobile, which almost consists dual SIM. One SIM from the Airtel and second SIM will be from the Jio. If one network will be affected, automatically our mobile will move on the second SIM or Jio. That's why we can utilize either one potent way SIM first, or we can go for alternate SIM second. In a same way, hybrid plants are the combination of two best parental plants, either on disease resistant or anything else. It is generally called open pollinated types or OP techniques to make cross of desired pollens. I have told you we have to just select the best plant. Just keep on my words. We have to select the best plant either on the phenotypic basis, which was as in conventional breeding, or we have to select the plants upon the basis of genesis. That is the main motto of classical plant breeding programs, in which we are going to select our plants upon the basis of their genesis. If parental plants are found more efficient, then definitely progeny will have the more efficient traits of economic importance. The main objective of improved quality, such as increased nutrition, improved flavor, growth, and height. Increased yield, abiotic stresses. Abiotic stresses means like temperature tolerance, hot weather tolerance, disease resistance, like the plant which grow nearby sea coast, that is called salinity, because seawater consists the pH of more than 8.4. That's why certain ions are going to affect the plant, as we have shown in yesterday's lecture with Dr. Sunita Kataria ji, who have given their talk on salinity effect on the plants. In the same way, I am going, salinity is going to harm our plant. That's why we have to produce resistance against salinity, extreme hot, and drought resistance by selecting the plant which is already surviving themselves under these conditions. If we are going to select that plant which already having the resistance against salinity, extreme hot, temperature, heat tolerance, etc., then definitely their progeny will be more resistant about abiotic stresses. In a same way, these traits are going to affect the socio-economic condition of poor. If farmers have more productivity, then definitely they are going to enhance their socio-economic impact. In a same way, these are the certain picture which I have taken. These are the certain plants in which we want to make process by just collecting anthers and pollens. We have just making matting, or we have just produced hybrid seeds. You can see the picture is telling itself. Now we can produce F1 generation, F2 generation, we can make test cross, we can make back cross, etc. Test cross means when we are going to confirm the identity of a particular allele existence in a crop. Back cross means we are going to cross F1 generation again with the previous one, with the recessive traits like eye color, small red, or flower color, light colors. That is called backcross, in which we are going to mate F1 trait with recessive traits from previous F1 generation. You can see the ratio, you can see the results in which we are going to produce hybrid tomato. Just you can see this tomato. Just you have to remember Mendelian genetics, in which one trait will be more dominant, 50%, ratio will be 25, 25, and finally 20, 25. Clear? So these are the three dominant traits for a color, while one become recessive, just in a Mendelian fashion, because we are relying on the genetics. Just you can see these are the colored flower which we have produced by classical breeding programs. Now, though these techniques were utilized earlier, but even consist certain limitations. First, the breeding can only be done between two plants that can sexually mate. We can use such breeding programs only in the family or only in the plants which having almost same genre or which can be meet. These limits the new traits there can be addressed to those that already exist in the particular species. We can only improve the traits which already present in their parental cell. We cannot inculcate the new desired traits. Just keep on my words. What I mean, I simply mean we can enhance only those traits which reside in their parental traits or only can be propagated into their F1 progeny, but not all traits can be modified. This is the great, this is the big, this is the very, very unfortunate limitation in certain plants. Second, when plants are crossed, many other traits also transferred along with undesired trait. Suppose we want to enhance the height of the plant, but simultaneously the color of the plants, the productive efficiency of the plants, the other traits are also going to propagate in their next generation, generally which we do not want. But being a limitation, such type of limitations, we cannot minimize the undesired traits. If we are going to propagate any desired traits like disease resistance, then other traits will also propagate in that F1 or next generation. That is the major drawback of this breeding program, in which we are also going to attain desired traits as well as undesirable traits which may cause problems to us. Now, next. Now, what is the need of the study? Because our population is going to increase day by day and our fertile land is going to shrink. That's why the agriculture land area is going to shrink, going to limited. That's why we are going to suffer with the demand of nutritional food, feed, and higher productive, and our cropping system. Higher traits are almost going to suppress. The extensive use of these chemical fertilizers also causing infertility, erosion, disturbing the pH, and physical and chemical characteristic of the soil are going to suppress or alter due to the extensive use of chemical fertilizer. That's why our soil is also going to affected. Just the example, Punjab and Rajasthan. Punjab and Rajasthan were the two states where wheat production was on the maximum height. The people from Rajasthan used extensive chemical fertilizers. That's why they become desert now. Because when we are going to utilize the chemical fertilizers, they are going to produce certain unsoluble salts of sulfate, magnesium pactates, etc., which are found unsoluble. That's why the pH, the physical and chemical characterization of the soil is going to change, or finally they become in a desert. Now the next number is Punjab. Now Punjab has started the utilization of more maximum number of chemical fertilizers in their crops for producing more and maximum productivity. But simultaneously, they are also going to harm the soil due to the extensive acidity and basicity of these chemical fertilizer like ammonium sulfate, DAP, etc. So the concrete strategies are now required for enhancing the qualitative and quantitative traits of crops to fulfillment the demand of increasing population. Now, how the productivity of agriculture crops may enhance? It is the question of believe question in a right scenario. How we can enhance our traits of economic importance, basically productivity. Now, is there any alternate pathway remain for us? और जैसे कि हम लोग आज के पढ़ाई के टाइम में आजकल हर चीज में चलता है, हो जाएगा, कर लेंगे, हो जाएगा, या फिर वी आर जस्ट गोइंग टू लीव द पॉपुलेशन इन अ डिलीमा कि सब चलता है ना, सब नहीं चलता है. दैट्स व्हाई वी हैव टू थिंक द कॉन्क्रीट स्ट्रेटेजी एंड द आंसर इज बायोटेक्नोलॉजी. नाउ biotechnology is the answer for every problem which we are going to face in our daily or routine life. What is biotechnology? I think students know better biotechnology kya hai, because they are doing BSc and MSc biotechnology, or they are studying the biotechnology subject. In my words, from myself, biotechnology means utilization of biological and engineering principles to enhance research and productivity by using microorganisms and their modified derivatives of human welfare like genes, toxins, ephelotoxins, etc. The advancement of biotechnological tools have been widely employed for the laboratory research, industrial production, medicines, pharmaceutical, healthcare. I think here MSc chemistry students are also present, so they better know how we can employ biotechnology to enhance the quality and quantity of our pharmaceutical drugs, healthcare, agriculture, animal husbandry, environmental, etc. Food as well. Now, we have divided biotechnology into 3 or 4 categories. Red biotechnology deals with the genetically modified organism, generally known as GMOs, like transgenic pig, transgenic cow, and transgenic sheep for maximum meat productivity. While while white biotechnology, manufacturing of useful chemical products on industrial scales like alcohol, butanol, chloroform, methanol, etc., which is going to use widely in the pharmaceutical drugs. While green biotechnology means the utilization of agricultural environmental biotechnology for more productivity, higher tolerance to environmental stresses and pathogens. We are just going to produce the disease resistance crops. We are going to produce transgenic papaya for edible vaccines. We are going to produce molecular forming, in which we are going to inculcate the desired traits by introducing desired gene in our products like papaya, that is also called edible vaccines. Now, these are the certain biotechnological tools and techniques which are widely using right now for higher researches like recombinant DNA technology, genetic engineering, PCR amplifications, molecular marker technology like RAPD, SNP, SSR, which is I am going to explain later, blotting, gene cloning, gene sequencing, germ plug conservation and gene banks, antisense technology and plant tissue culture, etc. These are the certain tools which are widely utilizing for enhancing our agriculture, our environmental, our healthcare, etc. by using such type of more efficient techniques to enhance our productivity in terms of agriculture biotechnology. Now, recent advances of biological sciences. What type of advances we are going to utilize? First of all, Gregor John Mendel experiments, that is also known as Mendelian genetics, with plant hybridization. I have simply told you we are just going to mix two best plant for producing best progeny, for establishing the law of inheritance which was given by Gregor John Mendel. The basis of genetics which stimulated researches by many plant scientists to improve crop production through molecular breeding. Modern plant breeding is applied the principles of genetics, but simultaneously they are also going to utilize the recent tools of biological sciences like molecular biology, cytology, physiology, pathology, biochemistry, plant biotechnology, statistical analysis, molecular markers. I have told you RAPD, AFLP, then SSR, microsatellite, SNP, SSR, etc. These are the certain markers which are going to utilize for enhancing the selection program. That is the MAS. MAS means marker assisted selection program. I will discuss later. Now, the modern plant breeding use the principles of molecular biology for the selection of desired phenotype for efficient breeding program. The success of breeding program depend upon the fact that how much efficient strains and host we have selected for our selection programs. The molecular marker system known as marker assisted selection, as I have told you, MAS. MAS is the recent tools for selecting a desired plant upon the basis of their genesis, their genotyping, the existence of unique allele in their germ plant. Program which is a highly versatile tools in modern era of biological sciences. Just believe me, it is the boom in agriculture biotechnology if we are going to select the host plant through mass. I will discuss later these things. Molecular markers can map thousands of genes within a host plant within a very short time of interval, which allow plant breeders to screen huge population of plants for those that process the traits of interest. I think we have heard microarray technology. Microarray technology is nothing. It is a just chip in which all desired markers are found embedded on the surface of the chip. Just we have to pour the selected DNA from the individual which are going to screen. We have to just put the desired DNA into the wells. After 30 or 40 minutes, you are just going to uh, find your results upon the basis whether they have hybridized with the host genome or not. Simply, we can say if color is going to change, means they are a potent genotypes. While if the color is not changed, means they have no complementarity at all with the marker. That's why these are the screening methods which we can utilize for the selection of a best host plants. The screening is based on the presence or absence. If the desired allele is found exist in our host, then we can say it is a plant which having best traits, either disease resistance, either metabolic pathways efficiency, or we can say it has maximum growth. While if the particular allele is found absent, then we can say this plant don't consist strong or desired metabolic pathways or efficiency for breeding further. So in this way, we can just screen out either we have selected a best plant or we have selected the not bad, or we can say ki not efficient plant in a general language. Now, these are the, you can see the differences between conventional breeding as well as genetic modifications. Conventional breeding program, we have to select the best plant upon the basis of height. Just you can take the example of height. We have selected this plant which have maximum height, while the yellow color flowers which have more efficiency of reproducibility. Okay? In the same way, we have made both the plants. This plant consists virus resistance against abiotic stresses, while this plant consists higher yield efficiency. We have introduced, we have met both the plants. In a hybrid plant, you can see we have attained higher reproducibility, we can say productivity or viral resistance as well. This was the concept which was utilized for producing hybrids in 2000, after 2000 or up to 2010. But after 2010, we have used this technique that is called genetic modification. After 2010, or we can say after 2000, we have just selected the plants, we have screened, or we found, if we found that this plant consists the genes found responsible for S resistance, then we have to select this plant through mass. After that, we have screened this plant for the unique alleles which are going to reflect maximum productivity. These are the mass programs in which we have just screened our two plants upon the basis of genotype or the existence of genetic alleles in their germ plant. Either the plant consists viral resistant gene, or other plant or male or female plant consists good productive genes. Then we have made them plants, or you can say the both germ plant, the DNA of both plant become hybridized. Then we can say our plant would consist resistance and productivity as well. Here we cannot say surely whether this plant will consist resistance plus growth, because we have just selected upon the basis of phenotypic traits, while here we can say 100% that the hybrid plant would consist all resistance as well as productive traits, because we have screened this plant, we have produced this hybrid plant upon the basis of marker assistant selection program that is called mass. That is the revolutionary idea for enhancing the productivity of cropping system, in which we have used conventional breeding earlier like this upon the basis of phenotype, or simply now it is a modern time, and now the agriculture biotechnology depend upon the fact that how much efficient parental genesis we have for propagating these parental genesis upon the basis of molecular marker system. Now, next, now progression of mass program continue, which is now I am going to continue. Unique genotypes are found across the genome that code for a specific protein. Just believe me, it may be enzyme, it may be hormone, it may be metabolite, it may be growth factor for the regulation of traits which we want, either heights, disease resistance, etc. Simply we can say any traits is found depend upon the fact that either our host plant consists desired enzyme, hormone, metabolite, or growth factor coming from the gene from their genome. That is a simple concept. Gene, that is also called the concept of central dogma, in which we are going to gene, mRNA, protein, or enzyme, and finally we are going to regulate the metabolic pathways. DNA markers have enormous potential to improve the efficiency of conventional plant breeding via mass. If we are going to inculcate it, the principles of molecular marker technology in our conventional breeding program, then we can enhance the crop productivity through marker assistant selection program, in which we have to utilize a potent marker for screening the plants which consist better genotypes. Mass includes selection of desired traits through utilization of biochemical, in which we are going to select the plant upon the basis of enzyme or protein or growth factor existence. These are called biochemical markers which utilize either enzymatic catalysis or proteins or any chemical substance. While morphological, these are the markers which are found depend upon the size, shape, height, color of the plant. These are called morphological markers. And robust DNA, robust in the sense of more potent DNA marker system like RAPD, RFLP, SNP, microsatellite, generally known as SSR, etc., for the selection program through the identification of desired QTL. QTL means quantitative trait loci, in which we are going to identify the linkage group where gene is found associated with the desired trait. Simply we can say marker gene association. Molecular markers are each in recognition for almost possible phenotypes. Sorry. Phenotypes, allelic specificity, ability to screen in Mendelian way. They are going to segregate in the Mendelian law of inheritance. They are found polymorphic. They have maximum reproducibility. They are found codominant. That's why these markers play an important role, because they have a certain property, like they are going to follow the genetic principles. They have the maximum reproducible process, we can say, or we can say ability, in which they can be utilized again and again. Polymorphic means they having the ability to diagnose genetic diversity, this plant versus that plant, in which first gene will be present while second gene will be absent. Simply we can say either presence or absence. These can be described by these markers only. As we can say, I have also written one paper in 2016 on animal genetics. This is a reference. Now, what will be required if we want to inculcate mass program? Simply, we need a molecular marker which may be as per our desirement, either microsatellite, which are widely using right now, maybe SNP, just existence, unique alleles upon the basis of either present or absent directly, or we can classify the germplasm for the study of genetic diversity and selection programs. Simply, we need a molecular marker for our better recognition, identification, confirmation of desired unique alleles in our host. There are 5 main considerations for use of DNA marker in the mass. Simply, we can say why we are going to utilize markers, because these markers consist following properties: reliability as compared to phenotypic traits, where we have selected the plant upon the basis of phenotypes. That may be wrong perception, because we have just seen the phenotype, not the genotype. That's why our perception may be wrong. But in case of molecular markers, these are found reliable resources, because they are just going to hybridize with the host genome, whether the gene is found present or absent. That is called reliability. Second, minute amount of DNA required for the identification, confirmation of particular alleles. The technical procedure for marker assessment simple. We required scientific procedure which cannot fail. That's why we rely on the molecular markers. Fourth one, higher level of polymorphism. We can detect the maximum number of unique alleles in different plant species. Simply, we can say these molecular markers can identify a great level of diversity among the plant, whether this plant consists desired alleles or not. Fifth, cost effective. Certain people will not be agree with me if I talk about the cost effective. Yeah, I agree. Cost investment is found higher when we are going to set up the biotechnology lab first time. But believe me, when your cyclic regulation are going on, when you are going to utilize in a running cyclic mode, then the cost will be minimized. That's why I have written these markers are found cost effective, but initially we have to invest a lot for establishment of biotechnology laboratory, PCR, RT-PCR, computational system. We need electrophoresis gel. We have to make sequencing. These all require scientific input as well as cost. But overall, if we talk overall, then the cost is cost effective. These are found cost effective simply after establishment of your lab. These markers will be available only on minimal cost. That's why I have written cost effective. The most widely used marker in major serials are called simply sequence repeat SSR, which are found potent tools of mass which are widely utilizing in the abroad as well as in India. I will give you the example later. Or microsatellite. These assets are having the wide identification capabilities for a specific gene upon the basis of compatibility. What is the SSR? SSR is nothing. These are the just stretches of 9 to 11, 12 base pairs, which can hybridize the unique host allele. Either they are found present, then the result will be there. If they are not found, then the result will be absent, or simply we can say nil. That is the simple phenomena which take place during the SSR selection programs. But if we are going to utilize other markers like RAPD, some are found PCR based, like AFLP, like RAPD, like SSR, like SSMP, and some are found like RFLP, non-PCR based marker. Simply during RFLP, we have not utilized any PCR. Next, these are the simple PCR which I have used in my lab during my doctoral program.
Sir, excuse me. Sir, your slides are not moving, I think. Sir, please check once.
Now it is moving.
Yes, sir. No, sir. Not moving.
Now moving.
No, sir.
Okay, just wait. I am just going to share once again. Some technical glitches may be there.
Yes, sir.
Always possibility remain for enhance the technical glitches. I'm just going to share once again. Now you can see the slides.
Yes, sir.
Okay.
This is
Thank you.
first slide. Yes, sir.
Thank you very much. Thank you very much.
Okay, sir. Now it's moving.
Thank you very much for informing me. I was
Okay, sir.
assuming that everything is going fine.
Okay, sir.
Thanks a lot.
Okay.
I'm just coming on the slide. Here I was. I think I was in this slide. This PCR I have used during my doctoral researches at Central Avian Research Institute in Jitnagar, Bareilly, on which, but I have used such techniques, such PCR for chickens. But right now I am giving the lecture, my talk on the agriculture biotechnology. Doesn't mean principle always remains same. Either it may be going to apply on the animals or going to apply in the agriculture, but principle almost remains same. Now simply you can see this is a PCR instrument. This is the screen. These are the buttons which we have to set. These are the programs which we have to modify. These are the phenomena of DNA repeating, or we can say amplifying the DNA. We are just going to 4 cycles: initiation, denaturation, initiation, and finally extension. These are the programs which are widely used during the PCR amplification by using molecular markers. These are the laboratories in which mass program is going on through RT-PCR. You can see the computer, you can see the rate of amplification here in the curve. These are the basic phenomena or methodology in which mass have to be employed. First of all, we have to develop the population for the selection programs. Second, we have to identify or we have to find the QTL, or we can say the location of a particular gene on which chromosomes it is found located. That is called linkage group, linkage while. Why linkage? Because each gene having the capability to decide phenotype later. That's why it is called the group of linkages with marker gene association. In terms of QTL mapping, we have to make, first of all, we have to make the genome of the plant in which we want to use mass programs. Finally, we are going to validate the QTL. Either we have confirmed or we have prepared a very good marker efficient markers or not. If we have confirmed, then we have to move forward for marker validation. We are just going to testing for our markers. Either we have produced a good marker or not. This is the mechanism of mass programs. Now, mass-based selection program is found more beneficial as compared to earlier conventional breeding program, in which the selection was depend upon the fact that how efficient phenotype we have. But recently, all the breeding regimens, programs we can say, or central government programs we can say, are relied on the mass. Either we have screened our population through mass or not. The molecular markers are advanced tools for the assessment of biological diversity of plant species to low investment cost as against मैनुअली सिलेक्शन. अगर हम मैनुअली सेलेक्ट करते हैं तो हमें बहुत सारे वी नीड लेबर, वी नीड पर्सन, वी नीड मैनिपुलेशन, वी नीड restoring data. But when we are going to mass, no parameter is required, only just we need the gene from the simply we can say DNA or genetic material from the host plant. We need a PCR, we need an electrophoretic gel, and or we need to sequencing. But if we are going to with the conventional breeding program, then we have to store data manually, selection, reliable estimate through analytical statistical tool, expandicity and reproducibility as well. If we are going to utilize markers, then these parameters play an important role in the decision whether we have select a good plant or not. Other advantages are also there. To characterize the large number of germplasm, we can utilize, I have just given you the example of microarray, that gene chip consists 96 bell plate, in which we can identify 96 more efficient plants just in a 20 to 30 minutes. But if we are going for conventional breeding, we have to take 1 day for selecting the traits, for storing the data, for register the data, etc. But by using such recent tools, we can identify our potent germplasm just within 30 to 40 minutes only. We can identify the best cultivar identity, confirmation, and assessment of purity, whether we have select a pure germplasm or not. Assessment of genetic diversity and parental selection, even we can say this plant belongs to this family. If you people have visited any dairy or research institute, then certain tags are found in the ear of the cow, जिसको हम बिल्ला बोलते हैं, बिल्ला number. Clear? That is called tag. Tag means the particular accession number of a progeny upon the basis of their parental genesis. कि इस parent का progeny ये है, इस parent का progeny ये है, we can select. While we are going to mass also can be implemented in sheep, goat, cow, poultry, duck. As my PhD supervisor Dr. Misra told, in ducks we are going to utilize certain marker assessment selection in ducks, poultry, cow, etc. Marker assisted backcross we can produce for identify the particular genetic makeup of a species, how much dominant or recessive alleles we have through test cross or back crosses. Marker assistant program found beneficial in every livestock, either sheep, goat, cow, poultry, or plant as well. Now, these are the certain markers which have utilized in different agriculture crops. You can see we have inculcated almost 20 to 40 markers, 2,240 markers in rice. That is the burning crop right now. Burning crop in the sense of they are going to attract us. Okay, we have made certain modifications rice only. Clear? That's why we have made more researches in rice, wheat, maize, etc. Rice we have introduced large number of SSR markers. A reference you can see. Maize we have introduced 1669 total markers, SSR markers. In soybean we have introduced 597, cotton 217, sugarcane 200, wheat 193. Still, wheat is going to suffer with the researches because we are not going to we are not going to make our focus on the wheat. We are just going for rice because rice is widely used globally as a food resources. Groundnut, we have introduced 1110 markers, SSR markers, potato 31, banana 28, sugar beets almost 30. Clear? This is the summary in which we have introduced SSR. SSR means micro satellite markers, or simply we can say short sequence repeats in terms of micro satellite markers. We have introduced. These are the research summary. आई एम नॉट सेइंग कि इट विल चलता है, इट हो जाएगा, हम कर लेंगे. These are the references. These are the references which have utilized for enhancing our crops recently within a 10 or 12 years. Now, conclusion. If I am going to conclude, the current breeding programs need to continue to make progress through conventionally used breeding approaches. Just simply the mean is we have to utilize modern mass breeding regimen or selection regimen for enhancing our crops as compared to earlier used breeding technology. Because our population is going to increase, we are expecting more nutritious food, we are expecting more furniture, more wood to our daily routine life. That is only possible when we are going to utilize certain recent tools of biotechnology for enhancing our crops. Mass has greatly assisted plant breeders for achieving goal of better crop productivity. If we want to produce more better crop productivity, then we have to utilize mass like programs. The exploitation of mass in respect of conventional breeding program have a great impact on crop improvement. Simply we can say, if we are going to compare, the mass is found better, reliable and potent tools for enhancing our crop improvements. The high investment cost and much scientific knowledge of mass will continue to be a major obstacle for its adoption. Huh, certain problems are there. We need to biotechnology lab, we need to select the markers, we have to find out the QTL, we have to take the electrophoresis gel, we have to make sequences. But if we are going to set up the biotechnology lab first time, then further the cost may be minimized. The scientific inputs definitely required. We need scientists, we need biotech professionals, we need biotechnology programs. Clear? Development of new marker technology can potentially reduce the cost of mass considerably if we are going to find out new molecular markers like SNP, etc. That may reduce the cost. That's why we have our researches should be continue, our efforts should be continue for finding out the new QTLs and molecular markers as well. If the effectiveness of new methods is validated and the equipment can be easily obtained, this should allow mass to become more widely applicable for crops breeding program. If we talk Indian environment, if we talk India, biotechnology have just started to explore in India. But our agriculture universities like Pantnagar, like NDRI Karnal, like Bhopal, these are already working on this principle, but not on mass. They are found rely on the molecular breeding. But such type of institutes may provide the facility. They have provide more jobs to biotechnology professionals. They can provide to more scientists in these institutes for performing such more efficient mass selection based breeding program for enhancing our livestock, our agriculture system, agriculture crops, cereals, and etc. like disease resistant plant, etc. So government should emphasize such type of programs should be continue for the betterment of humanity as well. Now, thank you very much for patience listening. And you can ask your doubt, whatever you have. Just Ekta ma'am, please continue.
Thank you so much, sir, for nice and informative talk. Now, I welcome our keynote speaker, Dr. Ajay Kumar, sir. Welcome, sir. Now, I hand over mic to Dikshit sir to address our keynote speaker to the participant. Over
Ma'am,
to you.
ma'am, I will take just 30 seconds. I am going to stop
Okay,
the share my screen.
okay, sir.
I am just going to share my screen because I was busy in my lecture that I am taking some time. Just wait.
मेरा कब आएगा 2 बजे
हाँ
मैं उनका काम करूँ
जी 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 A very good afternoon to all of you present over here. Right now, we have our most respectable Dr. Ajay Thakur sir. Sir, I welcome you a lot, being our eminent guest. And I know you were injured, or I want to show your photographs, which I have. Anybody can see this photo? Ekta ma'am, are you able to see the photo?
No, sir. It is not visible.
Okay, actually, I want to say something about our respected guest. Actually, they become injured by slipping on the snow, or they were not in the position to take the session today. But on my request, they have came here and they are blesses us in terms of his lecture. First of all, I want to introduce my guest, Dr. Ajay sir. Just wait. Okay, I'm just taking. Sir, sorry, sir, for delay. I'm just going to.
No issue, sir. No issue. Please take care. No issue.
I'm just going to select a profile. Yeah, now I am ready. I am going to introduce my guest, Dr. Ajay Kumar Thakur, which are currently working as a senior scientist in Biotechnology Division at ICAR Central Potato Research Institute, Simla, as a senior scientist. They have joined DMR Bharatpur as a senior scientist in 2004 after passing ARS NET. Dr. Thakur sir is a very hard working person, but they have obtained his graduation, PG and doctoral degree from Dr. Y.S. Parmar University of Horticulture and Forestry, Solan, Himachal Pradesh. They have selected for Agricultural Research Services, ARS, in 2008 from the ASRB ICAR, which is going to recruit the scientists in agricultural institutes. They have published more than 50 research papers, review articles in renowned international and national journals. Dr. Ajay Thakur sir also authored one very good competitive book, Agricultural Biotechnology at a Glance, which may help the students which we want, like Dr. Thakur sir. Simply we can say scientists in agriculture institutes. Or this book will be more helpful. The name of the book, I'm again going to repeat, Agricultural Biotechnology at a Glance, which can help you a lot for crossing competitive examination like ARS NET. They have edited 3 books, contributed 8 books, chapters, and 23 popular articles they have written. They have developed high efficiency plant regeneration and genetic transformation protocols in a number of crop, including Populus silita, or I think P. dilito, Dis, Punica granatum, and etc. Dr. Thakur sir associated with Brassica juncea improvement programs using biotechnological interventions from last 14 years in ICAR Directorate on Rapeseed Mustard Researches, Bharatpur, Rajasthan, where they have joined as a senior scientist earlier for mustard crop. Or luckily, I have visited their lab in Bharatpur, or I was so happy by visiting their facilities on biotechnology, which they have shown to my students during the visit of their institute. They have developed a core set of molecular markers for Brassica juncea genomics and worked on germ plant characterization and association mapping of various agronomically important traits in this oilseed crop. Dr. Thakur sir have also been granted one patent from Indian Patent Office, New Delhi, and associated in the development of 2 high-yielding Indian mustard variety, Giriraj and Brajraj. One Govi Sarson variety, register number is AKMS 8141, and 5 disease-resistant Indian mustard genetic stocks for the farmers. Dr. Thakur have received many awards from the reputed institutes, ICAR, DVT, DST, etc., for their hard work during their laboratory researches and higher researches. They are also an elected member of Plant Tissue Culture Association of India, PTCAI, and selected members of National Academy of Sciences India, NASI. Sir, I welcome you once again. I am so happy that you accepted our invitation. Being injured during the Simla, you have joined Simla, and just after you have slipped on the snow and got injury in your hand, but still you are with us. Sir, once again I appreciate. I salute your willingness to speak in our international conference. So I am just going to invite our respected, more eminent person, Dr. Ajay Thakur sir. Please, sir, you have to continue.
Thank you, sir. Thank you so much. Am I audible, sir?
Yeah, you are audible.
So, thank you very much for introducing me. And in fact, it's a perfect honor given by Dr. Pratap, sir. I am regularly in association with Dr. Pratap. When he was in Agra, he was heading biotechnology department there. And he's a good scientist, a good teacher, uh, good scientist, good teacher. But other than that, he's a very good human being. So I really appreciate your kind gesture, sir. And I congratulate you for making an excellent presentation. And I also congratulate your eminent Christian College for organizing this mega event. And I will say that you people keep on organizing such kind of conferences so that we people keep on coming there and keep on sharing our knowledge and in fact keep on learning from other guys. So thank you so much for inviting me here. And as far as regarding delivering the lecture, it was your request, but your request is just like an order for me. So whenever you will order me, yeah, I have to come there, so no issue. So thank you so much. And Sabhi ko namaskar. Actually, in the morning, Dr. Pratap asked me to present at 11 a.m., but I was caught somewhere in some official issue. But I think that happened for the good thing only because uh, the topic I'm going to touch, it is applied part of what Dr. Pratap has just introduced to the audience. So I think now it would be easy for me to present my work there, and it would be easy for the students particularly to understand the things, because Dr. Pratap has already introduced the topic I'm going to cover in a very, very lucid way, very, very effective manner. So thank you very much for inviting me, sir. I'm sharing my slides.
Yes, sir. Please.
Actually, I'm using this platform, Google Meet, for the first time. So,
Sir, you have to just share the screen and entire computer screen. Then you
entire
will be able to present your slides.
entire screen,
The present now option. Then you have to select your entire screen.
entire
Uppermost.
screen.
Yeah.
Okay.
Then you will be able to just open your slides. These will be visible. Again, we have to start from the beginning. Just we have to share the screen. Then we have to select
Yeah,
your entire screen.
It's coming, sir.
No, sir. Yeah. Now it is coming.
It's coming, sir. It's visible.
Absolutely fine. Absolutely fine, sir.
Fine, sir. So you kindly stop me if the slides are not coming or if some correlation is missing. Okay. Well, friends, a very good afternoon to you all. Myself, Ajay Kumar Thakur, and I am Senior Scientist in Biotechnology. Recently transferred from ICR Directorate of Rapeseed Mustard Research, Bharatpur, Rajasthan, to ICR Central Potato Research Institute, Shimla. And I have not changed my address because I'm going to present my work here, but I have done there in Bharatpur, Rajasthan. So Rajasthan is the hub of mustard, and you can see the excellent field we have here in this view. And this is our rapeseed mustard germplasm field. So this is the way how we are maintaining them. And when flowering comes, so we keep on selfing them to maintain their genetic identity. So regarding rapeseed mustard group of crops, like our institute is one of the Institute of Indian Council of Agriculture Research dedicated for rapeseed mustard group of crops. So that is why it is named Directorate of Rapeseed Mustard Research. So this rapeseed and mustard, my topic is genomics interventions for Indian mustard improvement. I'm going to touch some general issues and then what applied part we have done there in my lab. So these rapeseed mustard group of crops, they comprise of rapeseed and mustard. So mostly people are unable to distinguish what is rapeseed and what is mustard. So they are basically wild seed crops of genus Brassica. But in rapeseed, these are the species which are compiled here, grouped here. They're having apetulated leaf. There is an absence of petiole there. And in case of mustard crops, they are having petiolated leaf. So this is the only difference. So in rapeseed, it comes Brassica rapa, Napus, which is Gobi sarson, Oleracea, vegetable Brassica, and Iruca stiva, that is Taramira. And in mustard comes Brassica zancia, that is Indian mustard. And mostly our northern plains, it is grown there as a major oilseed crops. Then Carinata, Karanrai, Brassica nigra, Torniforte, and Sinapis alba. And whenever we talk about, sir, I'm audible now? Hello? 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 Sir, I'm audible?
Yes, sir. Yes, sir.
Yes,
Yeah,
sir.
I'm audible. Okay, you will kindly interrupt me when some connection is missing. Fine? Okay.
sir.
So this is a famous, whenever we start any kind of genomics research or breeding research in case of rapeseed mustard group of crops, so we refer this triangle given by Nagaharu Yu, a Japanese scientist in 1935, which explains how thousands of years back these primary diploids, particularly Brassica nigra, Brassica rapa, and Brassica oleracea, they hybridized with each other, underwent chromosome doubling by spontaneous process, and they gave rise to these amphi-diploid crops. Among them, Brassica gentiana is a major oilseed crop of our Indian continent, and Nepus is a part of European continent and in Canada also. So it is gobi sasam which is being cultivated there, and in India also, in colder regions, particularly in Himachal Pradesh, in hilly region, and in Kashmir part of J&K, people are growing Brassica Nepus there because it is suited to cold climate. Now coming to the Indian mustard scenario, like in 2019-20, these rapeseed mustard groups of crops contributed around 24% of the total acreage and around about 24% of the total oilseed production as such of the total oilseed crops. And among them, Indian mustard, that is Brassica gentiana, is the principal crop which is being grown more than 80% of the RM acreage in the India on an area of around 6.02 million hectare. However, there is a very grim scenario, particularly for oil seeds, that in case of oil seeds, India is unable to produce the sufficient oil seeds from its domestic for its domestic purpose, and we have to get edible oil imported from various countries like Indonesia and Malaysia, particularly palm oil, to meet our edible oil requirement. And we every year we invest more than 68,000 crore rupees every year for the import of edible oil. So there is a need to breed high yielding varieties, high yielding, particularly good in yield and resistant to various biotic and abiotic stresses through biotechnological interventions or through breeding, or as far as it's molecular breeding interventions. And for doing any kind of breeding program or any kind of biotechnology program, particularly molecular breeding, there is a need of superior genotypes having good yield parameters resistant to various biotic and abiotic stresses. So we have a collection of, say, biotechnology, particularly genetic engineering, comes to play the role when we don't have any source of any donor source available in the cross-savable germplasm. Then we go for development of transgenics. Now gene editing is people are following that in a big way, but its results are still awaited. So generally in conventional program, we need to find out certain particular genotypes which are resistant to various traits, and then those can be deployed in various breeding programs. And we have to go for mapping of certain traits like resistance or oil quality improvement using various kind of markers, as sir has explained, like different kind of markers, particularly SSRs. They are very, very useful, and now SNPs are available. They are more in number. Now people are using SNPs also. So SNPs are very costly and everybody cannot afford that. Every lab cannot afford. So we are going for microsatellite or SSR markers. They are the ideal DNA markers for various genetic diversity mapping, genetic diversity kind of studies, and various trait mapping or gene mapping studies. So now, as sir has told, that we need to have SSR markers for various genomic studies. But how do we come to have SSR markers? Now, say in case of these SSRs are sequence-based markers, so we need to have genome sequence of that particular crop available first of all. That is a major thing. And if we don't have genome sequence of that, then say like in case of Brassica gentiana, before 2016 there was no genome sequence information. So what we were doing, that the other crops, other related crops of Brassica genus, which have been sequenced, so we can go for taking their markers and go for validating them in these crops and develop markers for Brassica gentiana. So Brassica rapa genome has been sequenced by Chinese in 2011. Then Brassica nigra genome is on the way by Saskatoon, Canada. Brassica is already sequenced by USA people. Nepus is coming, and Carinata is from Ethiopia. So for it, no marker is available so far. Only our lab has developed some SSR markers for Brassica carinata. Now, as Brassica gentiana is our major key oilseed crop, so very limited markers were available for that before 2016. And it is having very narrow genetic base, so the markers are not showing polymorphism. But after the sequencing of Brassica gentiana species Tumida genome by Chinese in 2016, and by Delhi University South campus, Dr. Pantel's team in 2021, Brassica genome sequence of variety Varuna is in public domain now. So we are having enough number of markers for that. Now there are certain opportunities in molecular breeding of Brassica gentiana, because you say like in our experimental farms, we are going to harvest around 4 ton per hectare yield of Brassica gentiana. But when it is grown by conventional farmers, they are find it very tough to reach up to 2 tons. So there is immense scope for improving the yield when we breed for certain biotic and abiotic stresses. And so far, around 40 linkage maps have been developed within crops, and various states, particularly white rust resistance, then oil content, then glucosinolate or erucic acids, they have been mapped earlier by various workers. So genome certain markers are available for them. Then now sequencing of Brassica genome Tumida and Varuna has been completed. So we can design markers for every chromosome, for every linkage groups, and we can go for trait mapping and validation. But there are certain challenges, as I have told, that the level of polymorphism in Brassica germplasm accession is very, very low because of the narrow genetic base. And whatsoever markers have been developed for various traits, they're not showing perfect kind of correlation, perfect kind of segregation to the next generations. So this is a major issue. And again, Brassica is an amphi-diploid crop. Its genome is having large-scale duplication. So these are the certain challenges by working on Brassica. And there are certain biotic and abiotic stress constraints which need to be targeted for Indian mustard crop improvement. Among biotic stresses, one is aphid infestation, we call it Champa there. Then Alternaria blight, then Sclerotinia stem rot. It is coming in a major way, and so far we don't have any germplasm available for going for breeding program. So this state has not been mapped yet, and we don't have any donor. Then for white rust, we have well-established markers developed by Delhi University South Campus. And this Orobanki, we call it Budia in local language of Rajasthan. It is coming in a big way. Among abiotic stresses, high temperature stress is a major issue when we go for uh, establishing a crop at seedling stage. Then salinity affected soil is there, particularly in Rajasthan, and Rajasthan is contributing around 50% of Indian mustard acreage and production. So it is a major area of Indian mustard. Then drought issue is there. And then quality improving, low erucic acid and low glucosinolate in seed meal. These are the major issues. And whenever we go for developing such lines, they are very, very poor yield. So these are the major constraints which need to be targeted. Now I'm coming to what I have done in last couple of years at my lab in Bharatpur. So as I told you, friends, that in case of Brassica genesiae, genome sequence was not available till 2016. So what we did that we took SSR markers from other species for which some markers were established or their genome sequence was available in public domain. We designed markers from those particular genome sequences, and then we evaluated their transferability to Indian mustard genotypes. We have taken 12 high-yielding and genetically diverse varieties, and those markers are showing amplification and were showing polymorphism there. So now we have developed markers using this cross-transferability approach to Brassica varieties. We could develop around 104 polymorphic markers. Then we are using certain wild species for making introgression of certain traits like Brassica tournifrutii is resistant to drought, then Fructuosa is resistant to aphids. So again, when we go for making crosses and when we go for studying the inheritance of those traits, we need SSR markers, particularly specific for those wild Brassicas. Like how could we detect like what kind of genomic segment has been integrated there in our hybrid? So we have developed markers for those wild species, and we published in Australian Journal of Crop Science. And simultaneously, because we were not much enriched lab at that time, and we were not having sequencer and all facility, and we were not doing any SNP genotyping work. So what we did that we submitted a project on development of a core set of SSR markers to SCRB, Science and Engineering Research Board, New Delhi. Am I audible, Pratap sir? Hello, I'm audible.
Yes, sir. You are audible.
Please
I'm
continue.
feeling like I'm speaking like and nobody's listening. So yeah, keep on checking me in between. So we
Okay, sir.
submitted that project that we will take around 1,000 markers from these species and use their evaluate them for their transferability to certain Brassica genotypes, which is a genetically high diverse set. Actually, we phenotyped around 4,000 Indian mustard germplasm in our field and developed a core set which is genetically highly diverse, and develop a mini core set using agromorphological traits, which comprised of around 46 genotypes, and that was representing the entire diversity of Indian mustard germplasm available in the country. So we used those 46 genotypes, and we evaluated these 740 markers with them, and found that 236 markers were showing polymorphism, and these 236 markers represent a good set of SSRs used for any kind of genetic mapping work or any kind of diversity studies or association mapping work. We recently published this work in Journal of Plant Biochemistry and Biotechnology, and now this set is available in public domain, and any worker who is going to who is planning to work on molecular breeding aspects of Brassica Zensea, this is a core set for him, a reference set for him, and I expect that in the coming time people will use it like anything because there is no SSR marker information. As I told you now that the Brassica Zensea genome is in public domain, again this exercise has to be done, that the markers have to be designed and they have to be validated to know their polymorphism. Then, as I told you that Naga, how do you establish that evolution kind of thing by making conventional crosses? We took species of all the Brassica genus of U triangle and studied SSR marker transferability there, and it provided us certain interesting results and provided insight into the origin and evolution of various Brassica species, like how could they originate in nature thousands of years back. And we could find out the genetic differences between these species, like in case of Brassica Zensea, it is a cross of Brassica nigra and Rapa. We found out that you see the genetic distance that Brassica Zensea is more closer towards nigra genotypically than Rapa. So we again validated this use triangle, and we found out what kind of how much genetic distantly, how much distantly these their primary diploids are from the secondary amphidiploids. So when we develop this dendrogram, and you see here that Brassica Zensea Indian mustard is coming along with Brassica nigra and Brassica rapa. Now, from this genetic distance, we could find out that it is closer to nigra than rapa. And here in this cluster, you can also see that Brassica Zensea, Brassica nigra is coming closer to Zensea, and rapa is quite distant away from it. But they are in one cluster. And all the species of C genome, like Brassica oleracea is C genome, Napus is AC genome, Carinata is BC genome. Those C genome species, they are having some genomic similarities, and they're coming in another cluster. And we published this paper in Hereditas, which is a Springer journal, having more than 3 impact factor. Then other emphasis, other target for biotechnologies given by the director DRMR, that we were the first to develop first-based hybrid NRC SP506 at for directorate. So there were certain issues in its genetic purity, and whenever we giving the seed to farmers, again some issues arose there regarding the genetic purity, whether the seed the plants they're coming, they're hybrid or not. So we developed a unique DNA fingerprint for this NRCHP506 using SSR markers. We screened all those markers with both the parents P1 and P2, and you see the genotypes like 1920, which are showing the presence of both parental bands. They are the true hybrids. So we published it in Indian Journal of Biotechnology. And whenever we go for any variety characterization or any hybrid purity test, we generally go for grow test in the field or in plastic petri dishes. And in case of hybrid, you have to go up to flowering stage. You have to wait when the flower comes, and only then you will come to know whether this is male sterile or male fertile, or whether it is a hybrid or not. So by these SSR markers, now we have eased the program, and we have reduced the time for the genetic purity testing. When we were in earlier studies, when we were not having a good number of SSRs with us, we used RFID markers for studying genetic diversity in germplasm lines of Indian mustard. And we were having 66 Indian mustard varieties then, and we characterized those varieties with SSR markers, and we developed a unique DNA fingerprint for certain specific varieties. And now our SSR marker set has been enriched. Now we have characterized 87 Indian mustard varieties and developed unique DNA fingerprints for 31 varieties. So now, say if somebody purchases some seed from DRMR, say of variety Giriraj, and he label it with another tag and starts selling it, with the help of that particular marker, we can detect whether that the seed material is of Giriraj or not, and then we can sue that person accordingly. Then we are also having Brassica rapa A genome species. You see that some people are growing. This brown sarson is obsolete now, but earlier, before 50, 60 years in India, only brown sarson was there. And then people later on shifted to Indian mustard. So this Brassica rapa is having 3 subspecies: brown sarson, yellow sarson is still grown, particularly in northeastern states, and then Toria is grown here also, particularly in Rajasthan. So we studied genetic diversity then, and from this diagram, you can see that from this dendrogram that KOS1 and KBS3, they are particularly brown sarson varieties, and they fall in between somewhere. And this is yellow sarson varieties cluster, and this is Toria cluster. So it means that yellow sarson, this brown sarson blood is there somewhere in yellow sarson and Toria. And we predicted the genetic structure of these Brassica rhodapa species, subspecies, and we published in Journal of Plant Biochemistry and Biotechnology. We were having around 1400 Indian master germplasm accessions, which were giving promising results for heat tolerance at seedling stage. We screened them again rigorously in field and in lab conditions, and we could found out 21 Indian master germplasm accessions, which were showing good degree of tolerance at seedling stage. So we thought to map, to go for genetic diversity characterization to identify certain genetically diverse parents to be used in breeding program, and then to map that particular QTL associated with heat tolerance. We are also developing simultaneously certain mapping populations like recombinant inbred lines for mapping of certain agronomically important traits. We are making one. We have made one Giriraj into Hira cross. This Hira is an exotic germplasm accession. It is very. It is having very minute size of seed, and Giriraj is bold seeded. So we are planning to map that bold seededness, and then certain other traits like it is having resistant to various diseases and having low erucic acid, low glucosinolates. We are also trying to map them with the help of SSR markers. Then we are having one genetic stocks, DRMR and IJ12-48. We found it resistant to multiple diseases, particularly for powdery mildew and stem rot. So for them, earlier no resistant germplasm was there. So we started making a real population for it for mapping of QTLs for particularly for my powdery mildew and stem rot. Then one genotype we got from some uh, other country, Poland. It is showing resistance to herbicide. So we have again crossed it with Giriraj and developing real mapping population for mapping of that particular QTL for herbicide tolerance. So now, as I told earlier, that there is a problem of orobanki. So orobanki gets killed when we apply certain herbicide there. But all the herbicide, mostly in Brassica agency, they are broadleaf, so they kill Brassica agency also. So now, when we are having certain QTL mapped for this herbicide tolerance, we can introgress that through marker system breeding into our promising Indian mustard varieties. Now those varieties will be resistant to that herbicide because of this QTL, and our orobanki will be killed. So this is the strategy. Now we have reached up to F8 stage, and now genetic mapping work is.
Sorry to interrupt.
Yes, sir.
Sorry, sir, to interrupt. Your PPT has stopped.
Stopped, sir.
Yeah.
Okay. Is coming, sir, now?
No, sir, please share once again.
Okay, sir. Okay.
Yes, sir. Now you can go. So please continue.
It's not visible to me now.
Okay. Sir, ignore. Please ignore.
Ignore, sir. Okay.
No. I think you have shared screen, but you have to share your entire screen.
So let me do it once again. Entire screen, fine.
You entire screen, or you can say window 2. I think it's technical error is there.
So I
Sir,
go to
sir,
my
please share one time window. A window you can share
so
one time.
just a minute. Is it coming now, sir?
No.
No, not coming.
Yeah. Yeah.
Yes,
Now
sir.
it's
Yes,
It's
sir.
visible.
coming. It's coming now, no?
Yeah. Yeah. Please continue.
So how can I go for slideshow now?
Slide. So just you have to take the. Just you can move forward or backward.
Okay, it's not moving.
It's not there. Not moving.
Not moving.
It is moving for you.
No.
No. Mhm. Okay. Sir, can you send me your PPT? Can you send me your PPT by mail?
I can send that, but it will take a lot of time.
No. Sir, please continue. Sir, please continue. Please continue. No issue.
Uh,
Sir, please continue. No issue.
it's not coming, sir.
Okay, sir. Please continue.
So I'm audible now. I'm audible, sir.
Yeah. You are audible.
Yeah,
Please
I'm
continue.
left with only 3-4 slides. So
Yeah. Please continue.
other than that, as Dr. Pratap has told, first of all, I must apologize for this
No
inconvenience.
problem, sir.
Must be some
No, sir.
internet issue, or maybe I'm
Please,
not
please.
able to operate properly. Sir,
Please, sir.
then we have, as I told you, that Delhi University South Campus has developed markers, SSR markers, actually IP markers, intron polymorphic markers for white rust resistance, tagging for white rust resistant and for low elusic acid and glucosinolate. So we were having varieties, Indian mustard varieties. Our institute is pioneer in developing a number of varieties there, and particularly our Giriraj variety is the. Sir, I'm audible now. Exotic genotype. And those exotic genotypes were having some linkage drag, and that's why whenever they used to come to end product of their in their varietal form developed through mass, they were lower in yield, and that is a major issue that we cannot give farmers any variety which is lower in yield. Of course, it is having resistance, but it is low. If it is low in yield, then farmers will never prefer that. So what we did that we screened our germplasm lines in field for white rust resistance, and we found one line which is DRMR MJB35. It was found resistant to completely resistant to white rust, and it was having two loci there. One loci from Hira and other loci from Donsk. It is very robust material for to be used as donor for marker sister breeding in Indian mustard. And then we started our marker sister breeding program, and at present we are in BC3 stage. We are using Hira and Donsk as specific markers for foreground selection, and as we developed SSR markers for those varieties early in our earlier studies, so we are using background markers for Giriraj variety to for the selection of recurrent parent genome. So we are at BC3 stage, and now we will go for selfing for 2 years, and after that we will subject our material to All India Coordinated Research Program improvement trials in different stations of the DRMR for multi-location testing. And if those varieties are quite good in yield and they are of course offering resistance, so we will release those marker sister derived varieties in public domain. So we also started some work. Actually, I am unable to show the slides. We have also started some tissue culture and genetic transformation work with Brassica agency NRC DR2, and we are doing genetic transformation with TBT1 and protease inhibitors in for alternative light tolerance. So our transgenic plants are in now T1 stage. We have characterized them for the presence of gene and gene integration with the help of PCR-based gene-specific markers, and they will be further characterized with my colleagues by my colleagues available there. And this is all about my presentation and what we were looking forward there. We were having a core set of SSR markers as we done in our DST project. Now we were planning to have we have actually synthesized design markers from Brassica agentia Varuna genome for all the 18 linkage groups of Indian mustard, and we have tested around 80 to 100 markers per genome in that diverse set. And now we are having around 500 SSR markers plus additional around 260. So we are having around 800 polymorphic markers in Brassica agentia, and this is a reference set for anybody in the country who is working on Brassica genomics. And then we are also doing certain association mapping work for tagging of various agronomically important traits, where we are going for genome-wide association mapping with the help of SSR markers. And our dream was to develop one high-density linkage map. So now, as our real population is in the final stage, so we will be genotyping that, and we will develop first linkage map of Indian mustard saturated with SSR markers, having markers on all the 18 linkage groups. And of course, we are doing marker sister selection for white rust resistant and quality improvement traits. And in future, in 3-4 years, we're supposed to come out with a product which may be released for the farmers if everything goes well. So, as Pratap sir has told in the morning that we published certain books, which I tagged here in my slide also. So, one book is Agriculture Biotechnology at a Glance. It is prepared by we prepared it based on our experiences of our last journey in last couple of 20-25 years till when I was associated in biotechnology, right from my MSc program. So we used to collect various question papers and all and prepare notes, and I have compiled them. And this is a very high selling book available at IRI New Delhi. Then we have edited certain books on crop improvement from CRC Press, then agriculture biotechnology from Springer, and Brassica improvement from Springer. In my last presentation, I would like to acknowledge ICAR New Delhi and ICR DRMR Bharpur for funding all my activities. DST, particularly Science and Engineering Research Board, for giving us that extra the startup research grant for young scientists. And my team members, particularly our mentor, Dr. K. Singh, who is a senior plant breeder there. He's a man associated with the development of Giriraj variety and development of first Indian mustard hybrid NRCSP 506. My colleague, Dr. Nanjundan, and my SRF and RAs, particularly Lal Singh, Vandana, Ankur, Nehanjali, Deepika, and all. So this is all about my presentation. Thank you so much. Thank you very much. And I do apologize that I couldn't show you all the slides. So I will do one thing, that I will, Pratap sir, I will mail my slides to you, and you kindly share the slides with all the participants if they are interested. Fine, sir. So thank you so much, sir. Thank you very much.
Thank you very much, sir, for your in-depth and knowledgeable sharing with us. And now the topic is open for discussion. If anybody want to ask sir, they can ask. If any student having the problem or having any doubt, they can directly interact to sir. Sir knows you. I will send the questions of my students to you directly by mail.
Yes, sir, we can contact anytime. No issue, sir.
Yeah. So, sir, I will. They will interact with you. Sir, thank you, sir. Once again, thank you very much for an informative.
In fact, I should thank you for giving me this opportunity. Thank you so much, sir. Thank you so much, and congratulations for organizing such a mega event. Thank you so much. Thank you.
Thank you, sir. Thank you very much, sir, for a kind presentation. Thank you very much, sir.
Thank you.
Okay, thank you, sir. Ekta ma'am, please, we can continue. Ekta ma'am.
Okay, sir. We start our second technical session, paper presentation. I honored session chairperson, Dr. Brajesh Dikshit, sir, Bundelkhand University from Jhansi. Shall we start, sir? Brajesh Dikshit, sir.
जी मैम स्टार्ट करिए सबसे पहले मैं आई एम वेरी मच थैंकफुल टू ऑर्गेनाइजिंग सेक्रेटरी Dr. Shivam Pratap ji who invite me in this conference as a session chair and thank you Shivam sir and ma'am you may proceed the session.
Okay sir, thank you sir. I hope that all the presenter are present. Every presenter have 10 minutes for presentation. Now, I invite our first presenter, Neetu Trivedi and Anuj Charles from MSc first semester. Neetu, Nitu, are you there?
Yes, ma'am, we are here.
Yeah.
Yes, ma'am.
You can start your presentation.
Okay, ma'am.
Okay, ma'am.
Okay, meanwhile, the students are preparing. Dr. Dikshit, thank you very much for accepting our invitation for session, the chair session. Thank you very much, sir.
This is my private privilege over here to attend this conference, and thank you very much for inviting me
I
for
know,
wonderful
you have certain
program.
issue. I think right now you are busy, but thank you very much for earning some time for us. Thank you very much, sir. Okay. 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 Please continue, Nitu. Please go ahead with the presentation, please. Nitu. Nitu.
Yes, sir. This is Anuj.
Nitu, you can continue.
Ji, sir.
No issue with timing.
Hello,
You can go. You can go efficiently. You have to explore yourself in a better way.
So, am I audible?
Yeah, you can continue.
Okay.
Please,
So,
dear
good
authors,
afternoon,
you
everyone.
continue, please.
Yes, sir. Good afternoon, everyone. My name is Anuj Charles, and I'm a student of MSc Chemistry, second semester. I, along with Nitu Trivedi,
Hello everyone, good afternoon to all. This is Neetu Trivedi, student of Christian Eminent College, Indore, doing my MSc in chemistry. And today, the title of the presentation is
repercussion of routine laboratory practice in the determination of degree of dissociation of weak electrolyte and deviation from the exposure of strong electrolytes. First of all, I want to thank Dr. Anubha Pandya for her guidance and Dr. Shivam Pratap sir for organizing this conference so we could do this today. Now, I would like to shine the light on abstract that we have prepared. The Oswald's dilution law, which was given in 1888, it is only applicable for weak electrolytes, but it fails in the case of strong electrolytes. This happens because the strong electrolytes are completely ionized on all dilution. This is happening because of a process called dissociation. This dissociation is a physical change. It happens as when an ionic compound such as NaCl, if we put it in water, the water molecules will surround the ions of NaCl and they will solvate the ions. And therefore, they reduce the electrostatic forces between them, which are quite strong. Under most conditions, ionic compounds will dissociate nearly completely. These are strong electrolytes, for example, hydrochloric acid. But the weak electrolytes do not dissociate completely. They produce relatively few ions. And when they are the examples are acetic acid and ammonium hydroxide. So this reduction in electrostatic attraction permits the independent motion of each hydrated ion in a dilute solution. This results in a disorder of a system when the ion change from their fixed position from the crystal to the mobile. In other cases, these electrostatic attractions between the ions and water molecules are so weak that the increase in disorder cannot compensate for the energy that is required for ions and crystals that are insoluble. Ideally, on very few dilution, we should get a certain activity coefficient, which should be equal to 1. But non-ideally, as we can see here, most concentration solutions arise principally because of ions of opposite charge attract each other. So when they attract each other, similarly, the ions of opposite charge repel each other. In consequence, the ions are randomly distributed throughout the solution. But that is not the case. That is only the case for an ideal solution. Now, in the present research paper, we have shown the deviation from the ideal behavior, which is concluded through experiments. I will introduce a paper today with the help of Oswald's dilution law. The statement given here is that the degree of ionization of any weak electrolyte is inversely proportional to the square root of concentration and directly proportional to the square root of dilution. As you can see here in the mathematical expressions here, when we apply the law of mass action, we will get the equation that we'll need to solve different mathematical problems. This Oswald's dilution law is only applicable for strong electrolytes. That is because they are irreversible. They do not have a proper equilibrium. So to get this formula, you can take an example of a binary weak electrolyte such as Ba. Here, as you can see, and alpha is the degree of dissociation. And you can see here the concentration and molar concentration, which is given in mole per decimeter cube. So when we apply the law of mass action and we put in the values of the concentrations, we get the formulas in the first and second equation. But as I told you earlier, that the weak electrolyte, the degrees of dissociation would be very small. As you know, the strong electrolyte will get ionized quickly, so they will have a higher value. But the weak electrolyte will not. So we have taken here 1 minus alpha as 1. So therefore we get this equation where the degree of dissociation is under root the product of K and V. Now secondly, I will explain the Corrosh law. The Corrosh law states that at infinite dilution, when the dissociation is complete, each ion makes a definite contribution towards equivalent conductance of the electrolyte irrespective of the nature of the ion. It means that at infinite dilution, when the dilution is increased at such a high level that it is irrelevant whether the ion is a cation or an anion. The conductivity will be given by those electrolytes only. The conductivity of ions of an electrolyte at infinite dilution is constant, as you can see here from this mathematical expression. Lambda infinity equivalent molar conductivity at infinite dilution. The C is for cation and the A is for anion. When the concentration of an electrolyte is almost zero, at this point the molar conductivity is called limiting molar conductivity. The molar conductivity of a solution can be defined as when we take 1 mole electrolyte and we put it in a solution which has 2 electrodes at a unit area of cross section, and the distance between them should also be zero. So when we decrease the concentration, the molar conductivity therefore increases. The increase in the molar conductivity is due to the increase in the volume that comprises 1 mole of electrolytes. So when we dilute it further, the conductivity will increase and the concentration becomes irrelevant at that point. It can be represented by EM. Now this explanation here is given for mathematics. The limiting molar conductivity is the summation of AX and BY, where lambda infinity is the limiting molar conductivity of an electrolyte that we have chosen. When a cation is the same in both the electrolytes, then the difference in the molar conductivity of 2 electrolytes does not depend on the cation. So when we take a chemical that we have used here, and the cation is similar, then the molar conductivity of the cation will be similar to the molar conductivity of the cation. There are two pairs of electrolytes with the same cations here. In this example, A and D in each pair. Then the difference between their limiting molar conductivities is not affected by A or D. This is the mathematical expression. Now we will move ahead with the materials and method. The materials we have used is the regular apparatus for any titration, the burette, beaker, stand, funnel, and the conductometer. Now the chemicals that I have prepared here is 0.1 normality of hydrochloric acid, 0.1 normality of sodium hydroxide, 0.1 normality of ammonium hydroxide, and 0.1 normality of acetic acid. All of them have safe strength, and the method that we have used is conductometric titration. Now the definition is given here. The determination of endpoint of titration by the means of conductivity measurements is a conductometric titration. This is an electroanalytical method that involves the measurement of electrolytic conductivity. Now the conductivity that is present in the beaker is measured by this conductometer. This conductometer measures conductivity in the SI unit of Siemens per meter inverse. First, what we did is we put 10 ml of HCl in 100 ml of beaker, and we filled the burette and attached it to a stand with an alkali. We have done 4 titrations, and the reactions I will show you later on. This conductometric titration gives a certain graph that we have used to conclude our experiment. Conductometric titration is based on the observation of changes in the conductivity. These changes are result when the ion's mobility changes. The mobility of ions that are originally present in the solution becomes quite different when they react with other ions. And so therefore, the graph is plotted against the conductance and volume of titrate, that is the alkali. On these graphs, we will see 2 lines. And when these 2 lines meet together, that is the equivalence point or the point of neutralization. Now, here are the reactions for each titration. First, the ammonium hydroxide reacts with HCl to form common salt, that is, we all know, sodium chloride, and water. This is the neutralization reaction. All of them are same in the principle, but we have changed according to the weak base and strong bases, weak acid and strong acid. The second one is ammonium hydroxide and hydrochloric acid, which gives ammonium chloride and water. The third one is sodium hydroxide plus acetic acid, gives sodium acetate and water. Fourth titration is ammonium hydroxide plus acetic acid, which are both weak, gives ammonium acetate and water. Moving on to the resultant discussions. Before the alkali is added, the active solutions are at high conductance. Means initially, before adding any alkali, when we put the rod of the conductometer in the beaker, we get the conductance of the acid, which is quite high. As we know, the strong electrolytes will dissociate quickly and they will give high conductance. But as we keep adding the alkali uniformly for every 2 ml, this conductivity decreases. Now, why does this happen? Because the highly mobile hydrogen ions are replaced by less mobile metallic ions. These hydrogen ions react with the hydroxyl ions of the base and they form water. So therefore the conductivity decreases. This also results in the neutralization. When an acid and a base reacts, they make a salt, and this process is a neutralization. Now when we keep adding the alkali, you will see that the conductance then increases after the point of neutralization. This is because of the highly mobile hydroxyl ions. Thus, the conductance is minimum at equivalence point. So when we plot the graph, we see two straight lines, and for strong base and strong acid, we get a V-shaped graph. Since both of them are strong, it does not matter whether we take the alkali in the burette or in the beaker. But when a weak base, like ammonium hydroxide, is added to a strong acid, first the conductivity will fall as before, but then it will remain stagnant. This happens because ammonium hydroxide is a weak base and it does not dissociate completely. So therefore, the conductance will be the same for some time. After that, it will decrease or decrease depending on the principle of Debye-Huckel. In the case of weak acid like acetic acid against a strong base, the conductivity first decreases into the formation of salt. Similarly, this is happening here when we take NaOH and acetic acid, the conductance will decrease because the salt is being formed, and then it will increase to a certain point, and then it will be stagnant because the acetic acid is weak acid. After the point of neutralization, the conductance increases more rapidly to the axis of hydroxyl ions because the base here is strong and we keep adding the base from the burette to the beaker. Now, when the acid is weak and the base is weak, the conductance increases first in the beginning of neutralization. In the titration of a weak acid and weak base, the conductance first decreases, but it soon begins to increase due to the salt formation and then finally becomes constant. Now, all this is happening because of the Debye-Huckel theory that we have used here to justify our results. This theory explains the increase in molar conductance with dilution in case of strong electrolytes. The previous laws that I had explained to you are only applicable for weak electrolytes that were given initially. And this theory has come after that is applicable to strong electrolytes as well as weak electrolytes. So when a strong electrolyte dissociates into a positive and negative ion, every ion is surrounded by an ionic atmosphere of the opposite charge. And this atmosphere is spherically symmetrical. But when we pass a current, when we use the cathode and anode and we put it in a beaker, in that solution, then this atmosphere gets disturbed. So the central ion of the ionic atmosphere moves towards the oppositely charged electrode. During this moment, it has to build an ionic atmosphere of opposite charge. At this time, the ionic atmosphere dies out. These two processes do not take place simultaneously. There is some time lag, and this time is the time of relaxation. During this time, old ionic atmosphere pulls the ion back, and so the motion is slowed. This effect is the relaxation effect or the asymmetrical effect. You can see here from this picture. Now the second effect that takes place is the electrophoretic effect. If the solvent is polar and solvent molecules move along the ionic atmosphere due to the ion-solvent interaction, then there will be some hindrance in that movement of the central ion towards the electrode. Dilution diminishes electrophoretic effect and hence increases the polar conductance of strong electrolyte. You have to remember that whether it is a strong electrolyte or a weak electrolyte, the conductance will increase as the dilution is being increased. From this picture, you can understand this. Now, I will show you the observations that we have made from the titration of NaOH and HCl. First, in the beginning, we had not added any base. So at 0 ml here, you will find that the conductance is very high. But as we keep adding the alkali, that is the NaOH, the conductance keeps on decreasing. So at the fourth serial number, you will see that the conductance becomes 0.39. This is the point of equivalence or the neutralization point that you will see here in the graph. Then after that, this conductance keeps on increasing, and this increases because of the hydroxyl ions only. It is not because of the protons that HCl is given in the solution. You can see the scale in the graph that I have made here. It is a V-shaped graph. Now, for the second titration we have done of a weak base and strong acid. Now the acid is in the weaker, and like previously, the conductance is very high. Then the conductance keeps on decreasing. As you can see, from 0.79 to 0.72, then 5.9, then 5.4, then 3.9, then 3.6. And then at the ninth serial number, you will see that it becomes stagnant at 3.2, 3.2, 3.2 again and again. This is because ammonium hydroxide is a weak base, so it does not dissociate. Therefore, the ions remain the same. The conductance is given as 3.2. But further on, there are some disturbances in the conductance. It fluctuates because the hydroxyl ions can react later on with the protons that might be available and a salt that is being formed simultaneously. Now, you can see here in this graph, the conductance decreases first, and then the straight line that you're seeing, it remains stagnant. The point of neutralization here is at 16 ml. So I hope you have understood what I have explained here today regarding a research paper. Now, Ms. Neetu Trivedi, my fellow student, will explain the rest of the two graphs.
Thank you, Anush. As per the data announced by the Anush, the third observation table is sharing by the screen is like graph between the strong alkali and weak acid, which is acetic acid, at the reading of 0 when no more amount of hydroxyl ions are added, the conductance is 0.16, which is little high, but it is not so high because it is acetic acid or weak acid, weak electrolyte. And then when we add 2 ml of hydroxide ion, which is sodium hydroxide, the conductance increases, which is 0.28. Similarly, at the 3.234 reading, the reading increases, but at the reading of serial number 16, the graph is sharp increase because of the equivalence point, and the hydroxyl ion get the increase because the whole H plus ions get totally neutralized, and these increase is showing due to the hydroxyl ion. This conductance is just because of the hydroxyl ion. Now, the fourth observation table, which is showing between the weak electrolytes, which are ammonium hydroxide versus acetic acid. First serial number, when the volume of base is 0, the conductance showing 0.15 as per milliliter. And when we added 2 ml of base, the conductance increases. And furthermore, the conductance increases, but at the reading of 8 ml, when we added a volume of base 8 ml in the beaker, in the acid, the reading remains same, which is 0.20, 0.20, and 0.20. These readings showing the graph in which is staggered line. Here are the equivalence point of the graph, and the reading remains same. After that, when we furthermore added the base, the reading mean same. Here the graph is not increased very sharply because of the ammonium hydroxide ion is very weak base, and it cannot ionize very strongly because it can ionize very partially. And here is graph between the weak base and weak acid. Now, these three graphs are showing the deviation from the standard one. Standard is between the strong base and strong acid. These three graphs are showing the deviation which are like this. Okay, then this is the research and this is a new thing for D by Huckel theory. It is an experimental proof of D by Huckel theory. D by Huckel proposed only the assumptions or theoretical basis, and we have tried to explain.
So I hope you have understood what we have explained here today. The deviation that is given from the ideal solution, the ideal titration, I should say, from HCl and NaOH by different combinations that we have taken. And we have justified our results with the help of Debye-Huckel theory. Thank you.
Thank you. Dr. Dikshit, please. Dr. Dikshit, please.
Very nice presentation given by. I'm audible, sir?
Yeah, you are audible. Please. Please, sir. Please continue. Dr. Dikshit, you may continue. I think certain issue is there. Okay, Neetu and Enos. Neetu.
Yes, sir.
Yes, sir.
Thank you very much, Neetu, for an informative talk on the different titrations and salt concentrations. Now the session is open for the student who want to discuss anything. Please ask your doubt, whatever you have. Yes, Vinay. Vinay, you want to ask anything?
Yes, sir.
Vinay, please. I think your voice is disturbing.
There is a network issue. Hello.
Yeah. Ask Vinay.
Very good afternoon to all of you.
Yeah, good afternoon.
Anosh and Neetu. Hello.
Yeah.
Yes.
Yeah, you're audible. Please continue.
Yes. I want to ask you a simple question that it is understood that you have been choose some combinations for your research paper.
Yeah.
Likewise, strong acid versus weak base and weak acid versus strong acid. What was the reason that you chose that kind of combination regarding to your experiment, and what is the need of that experiment? What is the
Using,
motive?
yeah. The motive is to show the deviation from the ideal behavior, that is from the combination of hydrochloric acid and the sodium hydroxide, which are both strong. And we have taken these different combinations
Yes.
because they are common and they give the appropriate result that we require.
Okay.
So I hope you have
Thank.
understood my answer.
Yes, I am satisfied. Thank you.
Thank you.
Anybody else who want to ask something from the Neetu? Anybody else? Okay. If there is no question, thank you very much to Neetu and Anos Charles for a wonderful talk on the titration. Now, Ekta ma'am, please continue for the next participant.
Now I invite next presenter for presentation, Diptimani and Neetu Yadav from MSc Chemistry. Diptimani and Neetu Yadav.
Thank you so much.
You can continue.
Okay. I'm going to give a presentation on research article. Here.
I'm going to give a presentation on research article topic, a claiming on empowering diversity in science, global women breakfast in chemistry. Why so few and why care with Miss Neetu Yadav. Our motive is to study the status of women, diversity in science, presence and need to empowering. The graph presents scenario of women diversity in science. The percentage of women varies considerably by field from 2.4% in engineering to nearly 20% in the life science. Even higher percentage of women are found in psychology 31% and the social science 30%. So why very few? For why very few? Because of gender biasness, family responsibility, social responsibility, discouraging environment. But I'm not accept this. For why should we care? For these reasons, Wood Conference on Women Education says for World Conference of Women held in Beijing in September 1995 had emphasized that no enduring solution of society's most threatening social, economic, political problems could be found without the participation and empowerment of the women. What does World Bank say on women empowerment? A recent World Bank study says society would progress only if the status of the women is respected and the presence of an women in the family would ensure education of the family itself. Education and empowerment of women are closely related. To sum up my part, I would say the world cannot achieve be sustainability without 100% participation of the women. Thank you. Switching to Ms. Neetu for taking further.
Thank you, Diptmani. It is well said. It is well said that if the geeky society will become the gatekeeper, then for both women and a computer science will not lead for anywhere. So here there are some of the issues faced by the women in the science and in the research field. The main is the STEM gap. The STEM stand for the science and technology, engineering and maths. On the work platform, women make only up to 28% of the workforce in the STEM field. In India, it is just less to the 14% in STEM are employed in the research and the developmental institute. Women in the STEM face job insecurities and not represented adequately. Here we have a graph which shows the biasness about the subject that in STEM the percentile is very, very low. So how we can overcome the STEM gap? We can motivate more and more females to pursue career in the STEM field. Career breaks for the women, such as maternity leave, need to be accommodated. Representation of the women at the senior level, seniority should be based on the total number of the years of the work experience for the female too. So here we have some of the famous scientists and the researchers whose absence make STEM field, as we can say, incomplete. Here we have some of the eminent personalities. They are the Madam Marie Curie. She won Nobel Prize in Physics in 1903 for the research on radiation and the radiation phenomenon, and the Nobel Prize of Chemistry in 1911 for the discovery of radium and polonium. Maria Goeppert Mayer won Nobel Prize in Physics in the year 1963 for the discoveries of the concerning nuclear shell structure. Elizabeth Blackburn and Carol W. Breeder won Nobel Prize of Physiology and Medicine in 2008 for the discovery of how chromosomes are projected by the telomeres and the enzyme telomerase. Frances Arnold won Nobel Prize of Chemistry in 2018 for discovering the directed evolution of the enzyme. Emanuele Capenti and Jennifer Dendua won Nobel Prize in 2020 for the development of the method for the genome editing. So here we can say there is no force more powerful than a woman determined to rise. Thank you.
Now it's open for discussion. Any questions from participant?
Deeptimani and Neetu,
Yes,
Yes,
sir.
sir.
Good afternoon,
may
sir.
I ask, good afternoon, may I ask why you are not involving some Indian women scientists in your presentation?
Sir, there are so many female scientists are there in India too. And in future, when we will get some of the detailed information about them, then definitely we will going to include them. We have some kind of
Presently,
plan.
we are, and our society is working for women empowerment and वो महिला सशक्तिकरण जिसको बोलते
Yes,
हैं.
sir.
So
Yes, sir.
I think you must include the name of the scientist which we are working in the field of technology and science and all the things, and they are Indians.
Yes, sir. There
Some
are so
old,
many in India too.
okay,
We will definitely going to include them in our future work.
because we are in that time, in that scenario, we are missing some Indian scientists who are working in the field of technology and science. So this is must to recognize some scientists, especially Indian scientists who are working in the field of technology.
Yes, sir. Definitely. We will do
Okay,
that, sir.
nice presentation. Thank you.
Thank you
Thank
so
you.
much, sir.
Thank you, Neetu, for an informative discussion and talk on the role of women in the scientist community. Thank you very much, Neetu.
Thank you,
Thank you,
sir.
sir.
Thank you so much, sir.
Please continue with the next participant.
Next presenter is Chandni and Sachin Upadhyay from MSc Chemistry. Chandni and Sachin Upadhyay.
Good afternoon, everyone.
You can start your presentation, Chandni.
Yes, ma'am. Good afternoon, everyone. My name is Chandni Pandey. I doing MSc Chemistry in Christian Eminent College, Indore. Today, I am going to present a research paper on the topic of water purification techniques for treating all possible contaminants at low cost. I will share screen. So first is abstract. The standard for drinking water quality is typical sets by
Uh,
government or by
Sach,
international standards.
Chandni, your
Yes, ma'am.
slide is not visible.
Okay, ma'am.
Yes, it is visible.
Okay,
Please
ma'am.
be continued.
Abstract. The standard for drinking water quality and typical sets by government or by international standard. The standards usually include minimum and maximum concentration of contaminants depending on the intent use of the water. The history of water purification water purification includes a wide variety of methods. The methods used include physical properties such as filtration, sedimentation, and distillation, biological processes such as slow and filter or biologically active carbons, chemical process such as flocculation and chlorination, and the uses of electromagnetic radiation such as ultraviolet lights. Second is introduction. The process of low cost water filtration project is to provide a design for a low cost electricity free water filtration unit capable of elimination the physical and chemical contaminants of water. The contaminants may be particular matter, dissolved mineral, or microorganisms. There are number of methods for purifying water, but those are not economically feasible for rural peoples. Purification of water is the most essential for living a healthy life, as water act as a major role in day-to-day life. Especially in the rural areas, the access to safe drinking water is crucible. Drinking water of contaminant water may lead to fatal diseases. Third is methodology. The main aim of doing this project is by using locally and naturally available materials which is used to for purification of water, that for domestic and drinking purposes. The process of the project is shown in the figures. This water filtration such made by plastics container which focus on cutting down the cost while maintaining filter efficiency by providing affordable water filter for the rural households. This will surely improve the people's quality of living and reduce the risk of any waterborne diseases, therefore saving lives. Literature's review, identification of water qualities, identifications of material, designs of water purification, ground state, lake water, tap water, pre-treatments through and post-treatments to result and conclusions. Fourth is objective, to identify material and design the water purifier, to test the water quality parameter, to compare the pre and post treatment of water quality parameter, to estimate the cost effectiveness. The filtration system contains several layers of material which are locally available at the water sample, like groundwater, tap water, lake water, collect from three different area. The different layer of identified material used in the model and show figure. The design water filter contain two high quality plastic container which help to keep the water hygiene, and also the material used inside it purifies water. The material are placed inside the small plastic container according to their properties, which is kept to top of the large container, and large container collects the purified water. The container are transparent, so there should be clear visibility of for filter process. Figure third show the design model and material placed inside the top container. Fifth is materials. Fine sand, these are naturally occur deposits of glacial which are high in silica content and low in soluble calcium, iron, and magnesium compound, which are very useful in removal of sedimentation. But here, this media is mainly used for removal of iron from drinking water. Sand filter reduce high quality water without the use of any chemical aids. Hence, they are the natural remedies to eliminate the impurities of water. These show the working models. Rice husk, the material which is considered as waste, has a high level of silicon as per uses of carbonized rice husk in water purification. Charcoal also known as a carbon. The activated charcoal are very often used to absorb odors and colored substance from gases or liquid. Activated carbon filter reduce or even removes the amount of hazardous organic chemicals, and hundred of man-made substances found in tap and groundwater. Pebbles trap particles within the water through physical means. Therefore, water strained through pebbles will still need further treatment to remove pathogens and other particles that are small enough to slip through the cracks between the grain to make it drinkable. Aggregates. The high quality of aggregates normally required is not also readily available from commercial production processing, which may be designed to yield and satisfactory general purpose aggregate at least cost. Cotton. The cotton is effective because most pathogens which are attached to particle and plankton, particularly a type of zooplankton called copepods, passes through any filter. Most cholera, bacteria, and other pathogens are removed. I will call Sachin Upadhyay to continue the topic.
Thank you, Chandni Pandey. I am Sachin Upadhyay. Result: the completion study quality of water before treatment, pre and after treatment when determining display exit changes in water quality and satisfies the emissions limited given by IS. Table number 2 represent the physical and chemical properties of all three water samples pre and post treatment. Cost evaluation of designed filtration is projected in table 1. Cost items: plastic container first 15 liter quantity price 130, plastic container second 20 liter price 180, rice husk 500 grams price 100, charcoal 800 grams price 50 rupees. Then the next topic of conclusions: water quality standards are studied and taken under the constructions to that water should be within drinking limits. Water should satisfy the standard. One, filtrate the properties of the material well studied and identified according to WHO and IS, who is IS categorize the materials and
Please conclude the
finance
session. Please conclude the session.
and material required for purification of water. The water topic of reference development of low cost water purification technique 2013 taken Agrawal VK and 2019 household item purification low cost medical armed forces India. Thank you. I give this project because it is low cost project and made in household items can easily assemble. Or the main reason of this project is no electricity use. Thank you, ma'am.
Now it's open for discussion. Any questions? Thank you, Chandi. Now I invite
Thank you, ma'am.
Mohini and Prachi for presentation. Mohini and Prachi. Mohini and Prachi, are you there?
Vinay, you want to ask something? You have raised the hand.
Yes, sir.
Yeah, please ask. Quick.
Mm, Sachin and Chandni. Am I audible?
Yeah. You can continue,
Sachin
please.
and Chandni.
Yes.
So Sachin and Chandni, what is the common contaminant present in the water which leaves an effect on the health of humans?
The main content impurities
These project we use charcoal, rice flask, and many other cheeses, cotton, वगैरह, cotton, then can purify easily water and drink. It's not a unpurified. It's purified water. Some some impurities are visible in this water.
Okay, I want to ask one more question. That hardness in drinkable water is relevant to health or not? I
Yes,
repeat.
it's yes, yes.
Am I
Its hardness is removed. Yes.
okay?
Okay.
I'm satisfied. Thank you.
Okay. Thank you very much, Sachin. I want to add one thing. There are also certain insoluble and soluble salts which are going to alter the physical and chemical properties of water. Yeah, great.
Yes.
Sachin, thank you very much for a wonderful talk. Dr. Dikshit, you want to ask something? Dr. Dikshit? Dr. Dikshit?
Yes, sir, please.
Yes, sir, please. You want to add one thing? You want to add something?
There is a noise. I'm not
Vinay, please
able
mute
to
yourself.
listen.
Okay.
Hello.
Please mute. Vinay, please mute yourself. Sachin and Bune. Yeah. Yes, sir, please continue.
There's a wonderful talk given by Sachin and Chandni. I think this kind of research is very beneficial for the society. And
Exactly.
con- uh, so, Sachin, there is a suggestion for Sachin and Chandni. You
Yeah,
can
please.
also include the data comparison in between the low cost technique which we are using in this research paper and the marketly available resources, that means RO and all the things, where market में जो भी available resources हैं, that resources and your data which you are purifying your water, low cost technique, you can compare the data and publish the paper in the research journals and the society and the conferences, which is very beneficial for you and as well as for the society. Thank you, sir.
Exactly. I have the same view. So thank you very much, Sachin, for a wonderful talk on the water sanitization and pollution. Now, over to you, Ekta ma'am, for the starting of valedictory session.
Now it's time for valedictory session. I welcome all the dignitaries, honorable principals, sir, invitees, colleagues, participants, and students in valedictory session of International Conference. I welcome Dr. Ashok Kumar Gupta, sir, CMD, Research Foundation of India. I welcome Honorable Rajesh Shah, sir, in valedictory session. Now, I invite Dr. Ashok Kumar Gupta, sir, to say a few words. Welcome, sir.
Namaskar, sir.
Good afternoon, sir.
Namaskar. Am I audible?
Yeah,
Yes, sir.
sir,
You
please.
are audible.
Okay. Thank you. Hello, everyone. This is Professor Dr. Ashok Kumar Gupta, CMD RFI. I feel honored to be invited to this valedictory session of International Conference on Challenges and Prospects of Biotechnology, Environment, and Agricultural Research. The conference aims to provide the most complete and reliable source of information on the recent advancements in the field of challenges and prospects of biotechnology, environment, and agriculture researches. The emphasis will be given on publishing quality articles, making them available to the researchers and the young scholars worldwide without any restriction. This webinar on challenges and prospects of biotechnology, environment, and agriculture research covers a broad range of topics in this field. The National Education Policy 2020 has given a clarion call for holistic value-based education to prepare our youth and develop our nation. Education is fundamental for achieving full human potential, developing an equitable and just society, and promoting national development. In this line, RFI is doing efforts for organizing such events online and offline. Now it's my privilege and honor to welcome all the dignitaries, especially our patron, Dr. Saurabh Kumar Jain, who has given the permission for organizing this beautiful international conference. I would like to thank Professor Y.S. Chauhan, Director, sir. I would like to welcome Dr. Rajesh Vyas, sir, Principal, Christian Eminent College, Indore. I would like to welcome Dr. Ajay Jain, National Coordinator, RFI. I would also like to welcome Dr. Manish Dubey, sir, who is President, Central India Board, RFI, and Dr. Sanjay Prasad, sir, who is State President, NP Research Foundation of India. I'm also thankful to international guests and speakers, Dr. S.C. Degan, Janano Skopi. I would also like to welcome Rania Lempo. I'm grateful to national guests and speakers, Dr. Supriya, Dr. Sudhakar, Professor Dr. Pratik, Dr. S.K. Mishra, Dr. Ajmat Alam Khan, Dr. Mahadev Kumar, Dr. Ajay Kumar Thakur, and all the dignitaries who has given their input in this beautiful international webinar. I'm thankful to the entire team of RFI and Organizing Institute. I'm also thankful to all who have given their input in this conference and all participants who made this conference a success. For more information, you may contact through our official mail ID info@researchfoundationofindia.com. And for such other events, you can visit our website www.researchfoundationofindia.com. Thanks for your patience. Thank you again. Over to you. Thank you.
Thank you so much, sir, for your kind words. Now, I invite Honorable Shah, sir, to share their view. Welcome, sir.
Very good afternoon to one and all present here. This international conference on challenges and prospects of biotechnology, environment, and agricultural research is jointly organized by Department of Biosciences, Christian Eminent College, Indore, and Research Foundation of India, that is RFI, and other partners. On behalf of Autonomous A-grade accredited Christian Eminent College, Indore, I, Professor Rajesh Shah, Vice Principal, heartily welcome all our distinguished guests in validity session of this 3 days international conference. I heartily welcome and thanks our renowned guest of this validity session, Dr. Ajay Thakur sir, Senior Scientist, Biotechnology, ICAR, Central Potato Research Institute, Shimla. And I also express my heartily welcome and sincere thanks to special guest and session chair, Dr. Brajesh Dixit sir, Bundelkhand University, Jhansi. I am also thankful to all the guests of this 3 days international conference. I would like to thank and congratulate Dr. Saurabh Jain sir, young and energetic chairman and CEO of Research Foundation of India. My sincere gratitude to founder member and CMD of RFI, Dr. Ashok Kumar Gupta sir. My very special thanks to my dear friend, Dr. Ajay Jain sir, National Coordinator RFI India, and his team of RFI for their constant support and coordination. I also congratulate all participants of this conference. My special gratitude to management of Christian Eminent College, our chairman sir, our director sir, our director admin sir, our principal sir for their constant motivation and support. I would like to congratulate Dr. Shivam Pratap sir, HOD Biosciences Department, Christian Eminent College, his department members, all members of organizing committee for this successful international conference. My best wishes to all. Thank you all again. Thank you very much. Thanks everybody.
Thank you so much, sir. Now it's open for feedback. If anyone want to give feedback, they can give their feedback.
Please
Hello?
continue your feedback.
Hello.
Anisha?
Yeah. Please
Yes.
continue.
Good afternoon to all. A very afternoon, respected delegates, guests of the validatory session, and members of the committee and organizing committees and various college. I'm Nisha Tomar from Dr. Institution and feel proud of it being a part of this international conference. Of aspect of agriculture researches. Between 3 to 5 March 2022. In past 3 days, we got opportunity for attending session on recent tools and techniques. We will definitely help us to our future prospect. And all participants learn lot of many things, and hopefully all are going to benefit from the conferences. I express my gratitude to the Christian Eminent College Indore and entire organizing team for the conducting such a wonderful interactive session to all of us. Once again, thank you for very much all of you. This valuable knowledge is quite helpful that your institution will organize such interactive session and continuously for all of us, which make us relationize towards the effective teaching. And thank you for organizing this wonderful session. Thank you once again, sir.
Thank you, Nisha.
Please next.
A very warm good afternoon to respected dignities. Nisha Dinkar, head of the Biotech Department, KCMT Bareilly, wants to say something. Actually, I am very proud, privileged, being a part of this international conference organized by Department of Bioscience. First of all, I would like to thank Dr. Shiv Om Pratap, Organizing Secretary, Professor Ekta Rawat, Convener, C.K. Tiwari, Sir, Technical Committee, and various head of the Department of Christian Eminent College. I would like to express my gratitude to all the extreme delegates of the webinar for their presence and contribution for making this webinar a great success. In last 3 days, we got opportunity for attending various sessions on recent tools and techniques, which will definitely help us for our future prospects. Hopefully, all the participants learned a lot of things and surely going to be benefited from this conference. I would also like to thanks to all the members of the advisory board present over here. Once again, thank you very much to all of you. I am quite hopeful that your institute will organize such an interactive session continuously for the betterment of to all of us. Thank you very much, sir, once again. Thank you very much.
Thank you, Nisha ma'am.
Hello.
Yeah.
Very good afternoon to all of you, the respected dignitaries, research scientists, scholars, and the guest of the valedictory session, and the member of organizing committee, and the various heads, faculties, Department of CEC, Indore, and Research Foundation of India. I, Brajesh Dikshit, feel proud, privileged to being a part of this conference organized by the Department of Biosciences, challenges and prospects of biotechnology, environment, and agriculture research, which is organized in between the 3rd of March to 5th of March 2022. During these 3 days, we got a lot of opportunity to attend the various sessions and techniques, tools, which is definitely helpful for the research scholar, for the society of our future prospects. All participants learned many things, and hopefully they are going to benefit from this conference. I express my gratitude to Christian Eminent College Indore and entire organizing team, especially the most hardworking and dynamic personality, Professor Shivam Pratap ji, and for conducting such a wonderful event and interactive session for all of us. Once again, I pay my cardinal thanks to all of you for this valuable knowledge sharing conference. I'm quite hopeful for your institution will organize such interactive sessions continuously for the society, for the scholar, for all of us. And thank you so much. Again, thank you so much for
Thank
the
you.
entire team. Thank you.
Thank you, sir. Thank you very much. Anything else? Okay. Ma'am, please continue.
Okay. I take this opportunity to present the report on challenges and prospect of biotechnology, environment, and researches organized by Department of Biosciences, collaboration with Research Foundation of India. I would like to summarize 3 days conference. In this conference, we had 6 technical sessions with 10 keynote speaker from diverse places, background and expertise. We had academician scientists from Lucknow, Jammu Kashmir, Bihar, Shimla, Greece, and from Indore. On very first day in 2 plenary sessions, we had thoughtful talk covering various aspects on research. On the first day, Dr. S.K. Mishra sir spoke about the advances in genomics and biotechnology of the next important alternative poultry. Dr. Supriya ma'am spoke about the challenges in agriculture biotechnology. Dr. Sudhakar Amble sir spoke about the importance of agriculture 4.0 for future prospects. Dr. Pratik Ranjan sir spoke about the challenges and impact of agriculture biotechnology of development society. Dr. S.C. Dragon spoke about the importance of soil management. Rania ma'am spoke about the impact of mankind. In second technical session, Dr. Azmat Khan spoke about the novel circular bioeconomic model for sustainable poultry product. In the second day of conference, we had two keynote speakers. Dr. Mahadev Kumar sir spoke about the overview of laboratory animal management and experiment. Dr. Sunita Kataria ma'am spoke about the application of priming agriculture for better growth and yield of soybean under salt stress. On the third day, we had a keynote speaker, Dr. Ajay Kumar sir, spoke about the genomic intervention for Indian mustard and improvement. We received more than 50 paper from different places. Overall, the presentation cover a wide range of topics as the conference is built with the intent of active and interactive learning. We sincerely hope that ideas and view shared during the interactive will show its seeds for sustained fruitful reset. Thank you so much with best wishes. Over to you, sir. Dikshit sir.
Thank you.
At the end of the session, it's time for vote of thanks. For that, I invite Shiv Om Pratap sir. Over to you, sir.
Thank you, ma'am, for our well wishes. A warm and graceful afternoon to all our most valued Honorable Chief Guest, Professor Dr. Ashok Kumar Gupta ji, Vice Principal, Professor Rajesh Sahasar, Chief of Session, Chair of Session, Dr. Brajesh Dikshit ji, and RFI Research Foundation. Now, it is my privilege to propose a vote of thanks and acknowledge the contribution of those persons who worked really hard for us for making our international conference Challenges and Prospect of Biotechnology, Environment and Agriculture Researches CPBEAR 2022 between 3rd to 5th March 2022 on a grand success. First and foremost, I sincerely thank to the Almighty for giving us the strength, the patience, the vigor, and preserve to achieve this goal. I want to extend my generous thanks to our honorable and dynamic chairman, Sir Samir Sarji, for their support and motivation for conducting our conference successfully. I am very thankful to our Honorable Director, Professor H.N. Gupta sir and Professor Y.S. Chauhan sir from the bottom of my heart for their continuous support and cooperation during the entire conference. Without such guidance and support, we would not be able to organize this conference. It is my privilege to express heartiest gratitude to our respected principal sir, Dr. Rajesh Vyasji, for showing me the way for preparing the framework of this conference to make possible it in reality. I am very thankful to our respected Dr. Saurabh Jainji, Chairman and CEO, Research Foundation of India, for helping us to conduct our event successfully by providing such a wonderful platform and versatility by which we able to conduct our conference. I express my sincere thanks to Professor Dr. Ashok Kumar Gupta ji, Chief Managing Director, Research Foundation of India, for their infinite support during the entire event. It is my most coveted occasion and great privilege to express my deep sense of respect and indebtedness to our respected Vice Principal, Professor Rajesh Sahasar, for his prudent thoughts, enlightening guidance, and creative suggestions, and giving me homely environment for conducting such a wonderful conference to all the delegates. We have we have been fortunate to have renowned identity from the academician, eminent scientific community for their wonderful interaction with the delegates in this international conference. I express my sincere thanks to national and international guests of the speakers during the entire session. Dr. S.K. Misra ji, Principal Scientist and in charge, Regional Central Avian Research Institute, Bhubaneswar. Dr. Supriya Kumara Velan, International Speaker, Communication and Leadership Coach. Dr. Sudhakar Umle, Associate Professor and Head, Mechanical Engineering Department, Sardar Patel College of Engineering and Science, Mumbai. Professor Dr. Pratik Ranjan Mungekar, Scientist and Professor, Global Educator, International Internship University. Dr. S.C. Dragan Jovanov Skopje, Scientist and Global Educator, Macedonia. Rania Lempu, Global Educator, and Dr. Ajmat Alam Khan, Professor cum Chief Scientist and Officer on special duty to Vice Chancellor. I am also thankful to Dr. Mahadev Kumar, Principal Scientist, Indian Institute of Technology and Research, Lucknow. Dr. Ajay Thakur sir, Senior Scientist in Biotechnology Division, ICAR, Central Potato Research Institute, Simla, and Dr. Sunita Kataria ji, Women Scientist, A Scheme of DST Fellow, DAVV Indore, and Miss Susmita Paul, Assistant Professor, Asian Royal University, Assam, for their valuable knowledge sharing and insights to the participant during the invitee talk and technical sessions during entire conference for the betterment of our youth participant. I am very thankful to the entire participants who have learned a lot, many things for their future endeavors, as well as the sponsors of this conference. I am sure that our college will continue to get significant provisions from the diverse group as the bunch of flowers for the ignition of our student further. I am also very thankful to all our head of the department and their faculty members for their remarkable presence and constant motivation for getting the pinnacle of success in terms of our National Conference 2022. I would indeed be failing in my duty if I do not express our gratitude to all the members of the organizing committee, Dr. Prerna Dubey, Professor Ekta Rawat ji, Professor Neha Joshi ji, Dr. Indra Dikshit ji, Professor Amit Nilose sir, and Professor C.K. Tiwari sir, and Ajay Bam sir and Yogi ji from RFI and other technical staff members from the RFI who help us a lot even on the adverse condition where technical glitches were there, they wholeheartedly supported us to make this program successful. It is not possible to thank everyone for such an appreciating involvement and their willingness to make our event successful by contributing their help directly or indirectly for everyone, which I forget right now. I express my sincere thanks from the core of my heart to everyone who helped us a lot. Once again, thank you very much to everyone present in this virtual meeting and valedictory session. Thank you very much from the core of my heart. Thank you. Over to you, Ekta ma'am.
Thank you everyone for attending the session.
Now it is a time. Now it is a time to take permission from our vice principal sir for making the end of the session. Please, sir.
Yes, sir. Thank you. Thank you. And I again, once I also want to thanks Dr. Anuva Pandya, madam. She is also session chair for our this 3-day international conference, and her contribution is very much appreciated for this international success of this international conference. So I express my sincere thanks to Dr. Anuva Pandya, madam, and her department members also. Thank you. Thanks, everyone.
Ma'am, I want to add one thing. Dr. Anuva Pandya has a lot help us for maintaining such a technical session as well. Actually, I forgot her name, but I am very sorry, ma'am. I am very much sorry to introduce. Or I am very thankful to all of you for helping me. Thank you very much, ma'am, for your appreciation. I am appreciating. Thank you very much, sir.
Okay, thank you.
Congratulations, congratulations, sir.
Thank
Congratulations.
you, ma'am. Thank you.
Thank you. Thank you.
Thank you. Thank you, Dr. Dixit ji. Thanks a lot for your wonderful session. Thank you. Thank you, sir.
Many congratulations to all of
Bioscience
you.
department, congratulations. Dixit sir, congratulations.
Thank you, ma'am. Thank you very much. It is possible just with the help of all heads and faculty members and speakers. Thank you very much.
Congratulations, sir.
Thank you, Vinay. Thank you, everyone over here. Now it is the time to disperse. Thank you very much. Indra madam, thank you very much for your kind support and help. Dr. Rasimi ma'am, thank you very much for motivating us.
Congratulations, sir.
Thank you very much to all of you present over here. Thank you, Prerna ma'am. Thank you very much for your kind help, support.
Congratulations, sir.
Thank you, ma'am. Thank you very much. Neha ma'am, thank you very much. हाँ मैम का नाम उसमें रह गया, संभव बोला था, मैंने बाद में ऐड कर दिया उनको। तो सबको नहीं, बीच में नाम रह गया था उनका, मैंने सबको बोल दिया था अबे हम डायरेक्टली, फिर मैंने उनका नाम अलग से ऐड किया बाद में, सर शासन ने बोला उसके बाद। एक्चुअली उनका नाम उस समय लिखना रह गया उनको सारे साइंटिस्टों के साथ में। थैंक यू वेरी मच मैम फॉर योर टाइम। चलो अभी तुरंत करते हैं। हाँ आ जाओ, आ जाइए सर। थैंक यू वेरी मच, थैंक यू मैम, आ जाइए आप, आ जाइए। ओके हाँ।